కనపడాలంటే కూడా అది ఇక్కడి నుంచే బెటర్లే. Good morning, everyone. So, how was your day yesterday? Okay. I don't know. I thought many students will be just mute कर दे ना उसको. Phone. मेरा phone. नहीं मेरा phone. Okay. Okay. So I thought many people would turn up in the morning, but looks like they did not even get up after seeing the Hyderabad. Okay. I am Dr. Sunita. I graduated from Kakati University from pretty long time, 1992. I've completed my MPharmacy. And after that, I appeared for Foreign Pharmacy Graduate Equivalency Exam and California Board Exam. And I'm a registered pharmacist at California and Nevada states of United States. I worked for 10 years as a registered pharmacist. And I returned back to India and I joined with a JNTU. Same same story, just like assistant professor. Then I become a professor and I'm a principal now to my Center for Pharmaceutical Sciences. Okay. This is my brief history. And I have published many papers and I have guided about 10 PhDs and 140 MPharmacy students and guided several projects and also refreshing courses and seminars like this. We conduct almost every year, 1 or 2. Okay. So that is my brief history. But what I'm going to speak today is biomimicry, the nature to the lab. What is biomimicry? You know, the bio means something mimicking. So mimicry, you already know that some people do the mimicry. Like, they follow somebody and either a favorite actor or favorite politician, they will do it. But in the nature, that biomimicry is existing. From that, we have to take the ideas to ourselves and produce a drug delivery system which is equivalent to what already nature has given to us. Okay, that is the biomimicry, nature to the lab. Okay, what is contents I'm going to speak is what is biomimicry, how the nanoscience, because I'm going to talk about nanoparticles, nanoscience can be implemented in biomimicry. And these are all examples I have taken from the nature, the gecko and moth eyes, butterfly, peacock, lotus leaf, and Tuscan beaks. These are all, I am taking them as an example, how they have formed and what we can take and what we can learn from them. Okay. Have you seen this slide before in your WhatsApp? It's a light off. Take it, Babu. You can turn off the light. Have you seen this slide anywhere? Nobody has seen that? Okay. What happened in pandemic when we had a 2021 and 2020 from March to until now, all animals are roaming freely. But here many students are wearing the mask. Even I usually wear mask whenever I come to the crowd. Why that is happening? Because that animals are free of all viruses. They never got any problem with what the nature has been done. But the nature, the pandemic, the coronavirus has come, we are unable to withstand. We have to take care of ourselves. The same environment is given to us and also for the animals. But animals survived pretty good, but human did not. But that's why we lost almost 1 million or 2 million people during the pandemic. Okay. Why this is kept this one? Because we have to learn so many things from the nature. And we need to survive through the nature. Then we can take their ideas and implement in our day life that will be helping for us. So most of the problems human experienced even during pandemic and before and after also faced by many organisms. Over the course of evolution, many of the organisms gained more effective living styles than humans. And the organisms that are alive today are the examples, successful examples for us to take them into our lives. Okay. And we can learn many things in the nature that can be implemented into the daily lives, especially in our drug delivery system. Have we, you know that? Who has invented the aeroplane? Wright brothers. How they invented the aeroplane? After seeing the eagle flying, and they observed why the eagle could fly and why we cannot fly. And they kept some wings onto the back of their backside, and they tried to fly, but they couldn't. And they put the the wings in the front and back and tail and everything. They try to fly, but still they could not do it. Then they thought they can make the aeroplane, which is a light-weighted, equal to the eagle, feathers, and equal to eagle wings. Then they could able to make the aeroplane. So because they invented aeroplane, now we can visit many countries. And you know, yesterday Aravindo person, they supply the drugs to 140 countries. How they are supplying? Because of this, they will have a cargo planes and they will send the medicines to everywhere. And during the pandemic also, India has supplied many vaccines to many countries. How they could do? Because of this. So the nature has given some idea to develop something that is useful for the humans. So you have seen, I think, the gecko or a lizard, or, you know, the one which will be crawling. Everybody, whenever, you know, they don't get frightened by the lion, but they will be frightened by the gecko. And they will be saying, no, 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 I don't want to go. There is a lizard. So, but when you observe this lizard, okay, close, can you close that? Yeah. So when you observe this lizard feet, okay, when you observe very closely, they are in the macro size, okay. And when you, with a high microscopic illumination, we can see the macro size. When we go inside of one finger, they are arranged in a meso, and still you go inside, they are micro. But once you observe a single feather of that is in a nanostructure. If this nanostructure is in such a way, they have arranged in the bottom of these feet, they can stick onto the ceilings very firmly without falling in. But if we implement this kind of nanostructures, we can make a delivery systems which can be delivered the drug even 365 days when you stick onto the skin. So that is the mimicking of this gecko climber, and we can able to make a nanostructure of with this kind of material, and we can incorporate the appropriate drug. That drug can be delivered for a prolonged period of time. Okay. And by looking at that gecko feet, we are able to make the biocompatible patches, biomedicine we have done. And this is the biocompatible patches. What we have done, some of the patches separate, but after 2 weeks or 3 weeks, they will be combined and form a single patch or single skin. Same thing with the ear, even with the heart. We can make these biocompatible patches of nanomaterial which can stick on to the any kind of areas where you have an ulcer, and we can have a healing also. Same thing, the biopatches we can make light activated, and we can make wet adhesive. We can make the biocompatible patches light activated, because once we put it the light activated, initially they will be stick on to the area, but once we activate the sticking nature and compatible with the actual skin and the biocompatible, then they form a bond. They form a bioadhesive bond. After forming the bioadhesive form, slowly what happens, they will be trying to combine each other. Then there will be a blood transfusion in between. Then they can work in a normal way. Same thing, we have a wet adhesive methods also. We have to, after sticking the patch, we have to make wet adhesion. After few weeks, the wet adhesion become normal and it become attached to the area where you have an ulcer or where you have a wound. Same thing with the biocompatible patches with the heart also. And gecko tape, okay? By making the same nature of your gecko feet, we can make a gecko tape. This gecko tape, when you make it and stick to the any wall, it is able to hold 1 kg weight of any material. So you can have this gecko type of material tape, and you can put any drug inside. So this drug can be delivered the medicine for 365 days also, like, nitroglycerin, or we can have isosorbide dinitrate, or we can have some progesterone patches, some everything, or we can have any kind of material that you wanted a everyday need can be kept it in the drug delivery system, okay? And same gecko feet structures with a nanomaterial, we can make a shoes. These shoes are helpful for the trekkers or mountaineers, because they have to go and climb. So they need to, they are going against the gravitational force. So they have to stick to the area. So this is the way they can have their bottoms of the shoes, and the material, which is the whatever made is with a nanomaterial, they can stick onto that, okay? And same thing, the example of this, the mountaineers are using, because they will be prevent the slipping and while you going up or while you going down. And same thing, you might have observed the spider feet, okay? Whenever you observe the spider foot, they will be making the web. And whenever you cleanly observe, they will move their feet, the front feet to the forward and the back feet exactly on the same area of the front foot. So they will be moving the front foot and the back foot to the same area. Again, front foot, back area. Why they do that? Because they have a sticky nanomaterial in such a way they will be able to move and reduce the friction to the web and their body. So that is the spider foot will be moving very hairy spider tools in the web. And this is will mimic us to make a material which can be small van der Waals forces, and we can make a nanoscale pulling. So the same nanoscale material, if you can implement, this is also can be useful for the trekking and also can be holding the material, okay? And you know that this is the moth eye. When you observe the moth eye, there are several tiny nanomaterial present on the surface of moth eye. Okay, whenever any material falls on this, immediately they will be washed out, because they won't keep. They will be whatever the smallest nanomaterial present here will be trying to reject the material whichever falls on it. So this moth eyes technology is helpful for energy saving anti-glare properties that we can develop, like a self-cleaning windows. You might be having lot of windows. You know that our Rajiv Gandhi International Airport looks very, very pretty. Why? Because they made with a nano silver material coating. So whatever the material or whatever the dust falls on it, it will go away with a single droplet of water that will be rolled onto the dust, and the dust will be gone through the water. So they have dust on any material which is present on that. That is an example of self-cleaning. Next one is we can make energy saving. The glass coated with a very thin nano material, films of vanadium dioxide, and during the cold period, stop the thermal radiation escaping from the room and prevents the heat loss. In case of hot periods, it prevents the infrared radiation inside to the house. These kind of energy saving technologies are used in the Kashmir, Himachal Pradesh, where they have a very cold weather and they need to keep them into the warm. This energy saving material is coated, and that is also made up of very nano material structure. Same thing, anti-glare. We always use, whenever we have a drive, we need an anti-glare glass. So this anti-glare glass also coated with a very tiny material, just like a spider web which has a feet. The spider web feet has it. So another one, the chameleon. We always make the people that, they change the colors, they change their attitudes with the people, and it has a beautiful colors. And these beautiful colors are able to give us an idea to make our screens beautiful, and LED screens, or we can make some even our laptop screens. All these kind of ideas has come from the this chameleon. This does not have a different colors on their body, only the arrangement of nano materials on their body. And whenever the light falls in it, the light reflection, the light absorption makes the different colors. So that kind of nano material we are able to arrange on our screens and have a beautiful screens also. This is a chameleon, will help us make into the beautiful. Okay, another one, this is a cicada. I don't know how many of you see that. We always call light purugu because it will give the light in the air in the night sometimes. I don't know how many of you have seen, but I have seen that one. The cicada, then the cicada fly has two wings. When you closely observe the cicada fly, they have a very tiny nanomaterials, very tiny nanomaterials on their cicada fly. Whenever any bacteria falls on them, whenever the bacteria falls on these wings, immediately these tiny ridges will pierce the bacteria and kill them. So they never get sick. They never get sick, the cicada flies, because the tiny wings have a nano ridge. That nano ridges, immediately if anything falls on it, they will kill them by piercing. Once they pierce, the nucleus will come out, the proteins will come out from the bacteria or fungus, they will die immediately. So from this, we can learn the cicada fly, we can make able to some kind of a structures which can be antiseptic or which can be antibacterial in a hospital settings. Okay. And the fish scales, everybody want to swim. But when you observe the fish never get wet. Otherwise it will die. If we stay in the water for 24 hours, our skin will become bloated and start smelling and become, putrefy and will die. But fish never die. Why? Because when you observe that on that, they have a hydrophobic layer and having a repelling oil onto the surface of this fish. And how they are arranged? They are arranged in a nanomaterial size. So this nanomaterial and rippling oil and a fish scale able to reflect, are able to not to take any water inside, not to permit any water inside to the fish at all. That's why the fish can be stay in the water forever. Okay, that technology we can implement in a hospital beddings. That technology we can implement for the mountaineers. That technology we can implement for army who are staying at the borders day and night in a cold and hot weathers. That kind of technology can be used for nature, from the nature. And this is another one, skeletal flower. This skeletal flower, this looks very bright in a sunny day, but when it is rainy, it looks like this. That does not mean it lost pigment. No, it is not. But what happening when there is a sunny days, the air liquid interface of the petals causes diffuse reflectance. Whereas that diffuse reflectance looks like a white color. But whenever the water or rainy season, they increase the light transmission through their petals. That's where they look the clear petals. So this is just, there is no pigment change. Only because of the arrangement of the material, they are able to have a diffuse reflectance or a light transmission in both of them. Why they are able to do that? Whatever the pigment or whatever the material arranged here on the flower are tiny nanomaterial. Okay. So same thing, by taking the fish scales and rippling oil, we are able to make the transparent surfaces, like we can make a material which can be useful for the many mountaineers, and we can use the transparent surfaces even in the hospitals or any kind of pharmaceutical companies. And this nanomaterial, when you coat on your windows or any material, the people cannot see us, but we can see the people. Or we can make the observations. We can make the even examination halls. All these can be implemented, that kind of technology. Okay. And another one, a peacock, a beautiful peacock. We always want to have one feather, always, but it looks very beautiful because the light which is reflecting or absorption onto the feathers, I will give you and give you beautiful colors, and that are all this material, whatever the material on the wings are made up of nanomaterial. Okay. You can see here, when you can see the nanomaterial, this is one eye of the peacock. Okay, this looks like, but we go inside, this is again arranged in a meso size. Still we go inside of one area, they are arranged in a macro molecule. Still you go inside, this is all nanophotonic crystals. These nanophotonic crystals are colorless, but when you see that, there are beautiful color here. Because they are arranged in such a way, the tiny materials are nanocrystals are arranged. Whenever the light falls on it, they will reflect and absorb in such a way you can have a beautiful colors. Okay. So, and another one is butterfly wings also. That is also same thing. And butterfly wings, you know that JNU has a very big area in Sultanpur. We have 120 acres of own land of the JNU, where they have about 20 acres of land. We have a solar power. Okay, this solar power will supply the current to the not only Sultanpur, for us also. So why they are taken that butterfly wings technology that they have taken, and the solar power and everything has able to implement, and whatever the material they have implemented will give you the energy saving and energy production. Yeah. So same thing when you observe the butterflies thing, same thing. One area looks like this. Still you go inside, meso size, micro size, and they become a nano size. So these all become nano size. When you are able to arrange this nano size, what happened? Able to arrange this nano size, you can have a very beautiful colors with a reflectance of the light. Same kind of technology, same kind of butterfly technology, we are able to assemble the layers of the colors, like a single layer of nanoparticle, another layer of nanoparticle, and go on like one by one, and we can able to make a beautiful fabric. But all material is nanomaterial. Okay. And same thing, butterfly wings, we are able to make the solar power. This was done by the JNTU itself. And you know that nowadays we are getting the LEDs is one of the major thing that we want to save the power. But this kind of technology has been implemented pretty long time, 30 million years ago, by one of the butterfly that will be emitting the light. So these fluorescent patches on the wings of this African swallowtail butterflies work in very similar way to high emission light emitting diodes. So this technology has been taken from this beautiful butterfly of Africa. And one more thing, these butterflies can mimic you to make a different solar batteries, sensors, you can make it, you can make the batteries, you can make several things by seeing the arrangement of the material on the onto the butterfly wings. That will mimic many of the material. And we can make the beautiful colors even in LEDs. And this is a joke by a small girl of butterfly. Look, mom, I am dry, even though I am in the rain. Why it is able to tell? Because the area on the surface of the butterfly is not only in a nanoscale, but they are in hydrophobic in nature. So when we go into the rain, we'll definitely get wet. We need a raincoats, we need umbrellas. But butterflies will not have any umbrellas or raincoats, but they're surviving pretty good. So why can't we have that kind of technology to us? So that way we can walk everywhere without anything, that need not to wear anything. So the same technology also implemented hydrophobic nature on the pretty silver glasses or structures where we have to be dust free. Whenever you have a coating of small material of super hydrophobic surfaces, if in any dust or any material falls on it, with one single droplet that will be rolling onto the bottom of that, and it can be cleaned, self-cleaning can be initiated. Okay. And same thing with a lotus leaf. Whenever you see the lotus leaf, whenever have a droplet of water, it will never absorb. It has to fall off. Why it is happening? Because it is made up of a Okay, it is made up of droplets due to nano-sized ridges and wax-coated hairs which are present on the lotus leaf. So by taking this kind of technology, we can make a clothing with a nano coating to protect from the stains. That can be used for the hospitals. That can be used for the sterilization areas. Okay. Same kind of technology can be implemented. Like, you will have a t-shirts, you can have a, see here, the guy is pouring the water on that, but there is no kind of absorption or no kind of wetting, because we are able to make super hydrophobic of nanomaterial onto the clothing. Same kind of nanotechnology also can be implemented in different PET bottles, where you will not have a kind of any absorption or leaching. And also we can have a dew, mountain dews, which can be free of any absorption of water. We can have a super hydrophobic nanomaterial coating on it. Same, if you think about it, if you able to make a material which has a self-cleaning, then you don't have a person to clean your car, or you can have a footballs, have a self-cleaning, need not to worry about the cleaning the footballs and buying a new one. And same thing with the rain coats, water repellent raincoats. So these are, why can't we able to make this by thinking about the nature which has given, like a butterfly, peacock, or a simple cicadia fly, that all can take into the account, can make them. So this is another one, a nanotube forest. This nanotube forest also developed by by seeing the namibic beetle, which is a beetle which is in the forest, and they have a small tiny ridges on the surface. These tiny ridges, what they do is, whenever rain falls, these tiny ridges will collect the water from the air or from the rain, and they store in the bottom of the area. That is called nano dew or nano dots in the normal small bottom area. And same kind of, we can make a carbon nanotube forest where we can collect the rain or atmospheric water and able to store them in there. And this can be used for the later. Like in your forest, sometimes you'll have a rain, sometimes you don't. That time we can use them. And same kind of technology has make the namibic beetle to dew banks. These dew banks, what they do, whenever the water is falling or whenever the rain is coming, these tiny ridges will collect the water and store in these dew banks. And these dew banks, whenever you require, you can open the tap and use that water. And that water is very pure. Okay. And toucan beak. You can see the beak is very long compared to the body. Whenever we are very long, we'll fall down in the bud. But toucan never falls. It stay very stable. The bird never falls. It will fly and do everything. Why it was able to do that? Because whatever the beaks are made up of tiny nanomaterial, and they are pretty light when you compare with the body of toucan. So because of this lightweight, they're able to survive for almost 80 years and flying freely. So why can't we use this kind of technology to carry a certain drug delivery systems and deliver the medicine for a prolonged period of time? Instead of taking heavy weights of polymers, heavy weights of material, and having the sustained drug delivery start with a high volume of material, why can't we explore with this kind of material and have a material drug or APIs to deliver for a prolonged period of time? And same thing, toucan beaks. When we implement this toucan beak technology, we can have automobiles when we are able to protect, and we can able to protect the passengers from the crashes. So that way, the lightweight material will not have any problem when you have some crashes. We can implement this kind of technology even automobiles, or we can use in the aircraft components also. And woodpecker. This woodpecker will hit the bark 6 to 7 milliseconds, 6 to 7 times millisecond. 6 to 7 times they do it within a millisecond. Okay. So are we able to do that? Can we move our neck 6 to 7 times in a second? No. But this woodpecker able to do it. Why it is able to do it? Because it has a physical characteristics of head includes spongy bone on the upper beak and extended hyoid bone, a tightly enclosed the small brain within the skull, and plate-like high-strength cranial bone. So this is the area. It is made up of bones and spongy material in such a way that beak can be hit the bark 6 to 7 times in a millisecond. And it is survives pretty good. And whatever it is done, it will be equal to 1,000 grams per 1 minute. That kind of energy it is producing. So why it is able to do that? Because of the spongy bones and because of the hyoid bones, which are made up of tiny material and able to flexible very easily. Okay. Like they work like a shock absorbers. So that is the way we can have that kind of layers we can implement, and we in our technologies and able to make the a car bumpers. We can make the car bumpers of that, and we can make athletic helmets that can be useful. And also we can use the collars with a medicated material. So these are all can be done by that technology, and we can implement the medication also in the collarbones to help with the as a neck pain or spine and all those things. Okay. And shark skin, when you observe the shark skin, same thing. That is also a tiny material, but we are able to make a suit with a tiny material. Then that will be useful for many people. For example, for athletes, mountaineers, and swimmers, and also many other people, that will prevent any absorption of water inside. And another one, whenever you observe, I don't know whether you observe the flagella or not, but this flagella moves its neck in such a way, 20,000 times RPM, 20,000 times it will be moving in 1 minute, the flagella. So same kind of technology, we can implement it to the pumps, proton motive pumps, or delivery pumps, or we can implement into the insulin pumps. These are all can be, this kind of technology can be implemented to deliver different drugs and drug delivery systems also. So this is about the biomimicry, that nature which has given us, and we can take the ideas from the nature and we can implement that in our daily lives. And as you are younger researchers, and maybe you might be doing your MPharmC, PharmD, or projects or PhD projects, try to work on nanomaterial. And in our laboratories, we do a lot of nanomaterials. We do with all drug delivery systems like liposomes, neosomes, self-emulsifying drug delivery systems. We do the nanomaterial, silver, gold, coupling with the natural products, and also for materials. We have all instruments in our department. If you needed anything, we are happy to help you, but we are unable to do projects of yours in our labs because of the space and all other things, but we can guide you. And thank you for attending this conference, and hope you have any questions I can take. Okay. If it's no questions, we'll conclude it. Thank you. Okay, she's there. Good morning. Then we will be starting another speaker, Dr. Verena Brand. She is a consultant, clinical trial logistics, and miltenile biomedicine, Friedrich Albert Strabe. And she is coming from the Germany. She is logging in Germany, and she is going to talk the personalized medicine, advanced therapy medicine products, and regulatory aspects. Thank you, ma'am. Thank you. You can go ahead with your presentation.
Good morning, everybody. Thank you so much for inviting me to this conference. I'm going to talk about ATMPs and regulatory aspects. First of all, this is going to be the topics of my talk. I will give a classification of ATMPs, then an example of tissue engineered medicine, an example of gene therapy, and then go more into details how the CAR-T therapy works and how Clinimax Prodigy can help with that, and talk a little bit about the R&D pipeline of mid-near biomedicine, ATMP limitations, and some regulatory aspects. ATMP can be classified into three main types. Tissue engineered medicines, they can help to repair or regenerate or replace human tissue. ATMPs are breaking new opportunities for treatments of disease and injury. This is a tissue engineered medicines. You don't modify any cells. Then there are somatic cell therapy medicines. They contain cells which are manipulated to change their biological characteristics. And they can, in that sense, that they are not going to be used for the same essential functions in the body. They also can be used to cure, diagnose, or prevent diseases. Then there are gene therapy medicines, where also CAR T cells belong to, because they contain genes which are not from the human body, which are manipulated. And these can help to therapeutic, prophylactic, or diagnostic effects. They work by inserting recombinant genes into the body. Usually they treat a variety of diseases, including genetic disorders, for example, sickle cell anemia, or cancer, or autoimmune diseases, or other long-term diseases. We use a recombinant gene to be inserted into the cell, and this gene is created in a laboratory. So for example, this chimeric antigen receptor, which I'm going to explain later. So this is one example of a tissue engineered medicine, which is produced by TTEC AG. It is called cartilage cell transplantation or autologous chondrocyte transplantation, because it's from the same patient and reintroduced into the same patient. So that's why it's autolog. We need hyaline cartilage in our knee, in the meniscus, to support the function of the body, but the knee is a joint which is often used up and can be easily traumatically injured during an accident. So in that case, there is a possibility to remove a small piece of tissue or biopsy. Use that biopsy to amplify cells in the laboratory, and then to reintroduce the amplified tissue in a 3D matrix, or also as an injection where just hyaline cartilage is injected into the injured knee, and then the cartilage is going to grow and combine with the human body cells and tissue at place. That's the easiest way of an ATMP. Then there is the other example I'm going to give about gene therapy, or also short CAR T therapy. This is a type of treatment where a patient's cytotoxic T cells are changed in the laboratory with the purpose that they will bind the cancer cells and kill them. But that can also be used for destroying autoimmune cells. First, we will take blood from the patients to get T cells. So the T cells are going to be separated. Then we will introduce a foreign gene material into the T cell with a chimeric antigen receptor. This receptor is meant to find the cells which are not wanted in the body of the patient. We can grow millions of CAR T cells in a laboratory and give the patient the cells back by infusion. What is the chimeric antigen receptor? This is a special receptor created in a laboratory that is designed to bind certain proteins or cancer cells. The chimeric antigen receptor is then added to the immune cells called T cells. These helps the T cells to find and kill cancer cells that have the specific protein that the receptor is designed to bind. These changed T cells, called chimeric antigen receptor, they are grown in large numbers in the laboratory and given to the cancer patients. Chimeric antigen receptor T cells are being studied in the treatment of the same types of cancers, also called CAR. So the retargeting of the immune cells can be achieved by inserting new genetic information into the patient's T cells via gene engineering. So there is, in the meanwhile, there is a second generation CAR T or even third generation CAR T therapy available, because we had to overcome several limitations of current options. Because first, as we will see in the next slide, first there were only few targeting antibodies in targeting receptors inserted into the cell membrane. But in the meanwhile, we choose dual and triple targeting to reduce disease escape mechanisms, to provide new treatment options. So the first disease target we had at Milteney Biomedicine is DLBCL, which is like diffuse large B-cell lymphoma. Here we can see how the chimeric antigen receptor changed from the first generation to the second generation and to the third generation. Co-stimulatory signaling domains have been added to newer generations of CAR T cells to improve their ability to produce more T cells after infusion and survive longer in the circulation. So more unwanted cells can be targeted and the disease completely eliminated. Another possibility are adapter CAR cells, which are an improvement of the conventional CAR T technology. With that technology, we can target multiple cancer cells and antigens to control their activity. This increases the safety and efficacy of CAR cells. What is the concept? We don't directly target a disease-associated antigen on a cancer cell. The technology is split into two components, the CAR T cell and the adapter. The adapter CAR will be only active when both components are available. The CAR cell itself is not engineered to recognize the target cell, but to dock at this, to be a docking structure which is present in the adapter. The adapter consists of the docking structure and a cancer-specific binding domain. When the adapter is bound to the cancer cell, the CAR will bind to the adapter, initiate the killing of the cancer cell. We can use different adapters, enabling us to target different types of cancer cells with only one CAR. And this simplifies the treatment process and expands treatment options. This technology allows the activity of the CAR to be controlled by applying the adapter at different dose levels. The option to switch the CAR's activity is not addressed by current treatments, but it is believed to be an improvement for solid tumors. So we are currently working on relapse refractory AML, acute myeloid lymphoma. So I want to show, like a small video, like a small clips of a video. And I think, okay. I think okay, wait. have to choose I have to choose my option how to change. And I forgot something to when I started to change. I think I have to choose audio Einstellungen. Wait, where is this? Ton freigeben. Okay. So now to do this. A minute. Sorry. It's always the Zoom, you have to. Okay. Okay, I think I'm. Why is this not showing?
With the Clinimax Prodigy T-cell transduction process, you can now generate gene-modified T-cells in a simple and fully automated fashion.
Yeah.
The system allows you to perform procedures such as sample preparation, T-cell
T-cell
enrichment,
enrichment,
stimulation,
viral transduction,
viral
expansion.
transduction, expansion, and the final formulation of the genetically modified T-cells. All the way the user is guided step by step through the process. Simply follow the instructions on the screen to connect the tubing set, buffer, and TexMax GMP medium. After the system is primed, the starting material and the Clinimax reagents are connected. The T-cell.
Okay, so so how does it work? Here you can see it is. I just wanted to show you a little bit here this machine how it is working. So we will take fresh or frozen blood from the blood leukapheresis. We will prepare a sample from peripheral blood mononuclear cells with an IPC control. Then there will be a T selection. The T cells will be separated. The T cells will be activated to be able to be transduced with a foreign gene. Then there will be the expansion, and then we have a final cell product. This process time takes about 14 days. There's a possibility also to cryopreserve the cells before to produce larger amounts. The possibility is to produce that either centrally or decentralized. When we produce cells centrally, we have a single facility and we can parallel manufacture a lot of cells, and we have a consistent product quality. There could be also other possibility to produce this decentralized, which results in reduced logistics and an easy tech transfer to optimize the production. So here we can see that the starting material are PBMCs, either fresh or frozen. And we can see that the amount of cells which are produced when it is the fresh blood is about 5 billion cells. And if we start from frozen material and a bigger capacity of expansion, we can end up with 20 billion cells. So this is like about actually I wanted to show another video, but for some reason it didn't come. So we'll just continue with the talk. So we have a new therapy for DLBCL, diffuse large B-cell lymphoma, in our pipeline, which is already in phase 2. This is a tandem CAR technology, which contains receptors recognizing 2 antigens, CD19 and CD20, which are found on B cells. The name is samtoceptin. Then there are other products in the pipeline against major B-cell neoplasm and mantle cell lymphoma. Another possibility is our second generation CAR T cells, monoCARs, which have only one receptor, which are expressing the receptor recognizing CD19. And currently we are also entering into, so this is like still in the preclinical phase, but we are entering into the first clinical phase with these 2 constructs. And this one is also the MBCAR 2019 against B-cell acute lymphoplastic leukemia. Is also a tandem CAR where we are addressing 2 targets, CD19 and CD22. And then there are 3 other products in the pipeline against acute myeloid lymphoma and glioblastoma. What are the limitations of manufacturing? We have seen, so this is the video I wanted to show. Okay, that's going to be on the next. So this is a citation of the video, which I'm going to show from the NIH, from Dr. Anthony Welch. What is the specifically the tricky part of ATMPs? That the manufacturing process itself is a drug product. So that can lead to cross-contamination from different samples, because several samples are produced in one lab at the same time on limited spaces, and we can't scale it really because it's personalized. So for each patient, the cells have to be produced individually. So we can only produce them in parallel, but we have to be aware that we don't mix up the cells. So we end up with low reproducibilities, and we need complex operator training. It needs extensive documentation for each individual patient, and we have to be very careful about the raw material, where and how it is sourced, and preclinical validation. It needs a strict quality control due to the variability of the raw material, because each patient's material will be different. So. So I just want to. Yeah. So there you can see again that the prodigy was used in, to produce a lot of cells at the same time. So how does advanced therapies fit into the pharmaceutical product life cycles? We have three different phases: research and development, marketing authorization, and post-authorization. In research and development, we need the support for advanced therapy developers. We need scientific guidelines. During marketing authorization, we classify the advanced therapy. We need to find marketing authorization procedures. And in post-authorization, pharmacovigilance for advanced therapies are needed. As any other pharmaceutical product, it has to follow GMP requirements, but there are always specific guidelines for biological active substances and medicinal products for human use. There are specific guidelines to manufacture them, and we need a qualified person and for each batch release. Then we have to follow GCP requirements, good clinical practice, good manufacturing practice, good clinical practice requirements. There are also detailed draft guidelines on GCP specific to ATMPs. And then we need to follow GLP requirements, which are also specific to ATMPs due to their specific challenges, which I explained before, that each patient has to be treated individually. There are different guidelines for ATMPs. And for biologicals, the drug substance will be the cells which are manufactured, but they have to follow the quality guidelines from ICH and the efficacy and considerations of ICH. They have to meet clinical safety and efficacy, and in case of ATMPs, tumor vaccines are also considered gene therapeutic advanced medicinal products. So the different pharmacopoeias in the countries in Europe, US, and Japan have to be followed. For marketing authorization, we submit an application first to classify the ATMP, and then to get procedural advice. There are several reflection papers how to classify ATMPs available. Even for marketing authorization, there are several guidelines available for a risk-based approach, for labeling and packaging, and for human use opinion. The notified bodies have to be consulted accordingly, and there are special dossier requirements in place. Pharmacovigilance for advanced therapies, because they're pretty new on the market, are very important. So they are also the good pharmacovigilance practices have to be followed. And there are again a specific ATMP specific guidelines in place. Because we have to take a specific follow-up approach and risk management of ATMP. Thank you very much for your attention. Now you can address questions if you want. Are you still there? Do you have any questions? Okay.
Thank you, ma'am. Any questions from the participants? No questions at all? Ma'am, thank you. Thank you, Dr. Varina, for connecting from Germany. Thank you, ma'am. Hello, can you hear me, ma'am?
Yeah, I can hear you. I can hear you.
Yeah, looks like the audience didn't have any questions. And thank you, ma'am.
Because actually, there are like could be also side effects from like taking ATMPs that but we don't. They are still in process to be developed. So yeah, I thought there might be some questions for that, but anyway, it's okay. Thank you very much for inviting me. Thank you very much for being a participant of the conference, which is very interesting.
Thank you, ma'am. Thank you. Thank you. And we're going to have like a tea break in few minutes. Before we go for the tea break, can anybody wants to speak anything? Anybody wants to talk anything or want to express anything? No? Can you please guys come? At least you can introduce yourself. Come on. It's okay. No, nobody wants to. Yeah, come. Please come. 1 or 2 people, please come. Anyone? Last bench? Please come, no problem. There are not many audience also. Please come. Yeah. Can that have a table?
A very good morning to all of you. Myself, Dr. Pravakar Reddy. I'm a professor in working as a professor in pharmacy college. First of all, thank you, madam, for giving this opportunity. So, one thing I want to tell on this occasion, that this conference, what we are conducting, this is to know the development, what is going in the pharmaceutical industry. If you see the yesterday's strength and today's strength, so we see almost all the students, those who are having the presentation, only those students are present. But the point here is that we need to understand here, this conference is basically conducted for the budding pharmacists to understand what is the development going in the all over the world. And whatever the presentations are given through the various countries like Germany, yesterday we have seen Malaysia and other things. So we need to understand that there's a lot of scope is there in the pharmaceutical research. So this is a one type, one way, it's a motivation for all the students to understand that what are the research going in the pharmaceutical industry. And by keeping in view that, we should also, if you work in that area, definitely, as yesterday, we have seen the director of Arvindo sir, he also told that we are focusing not only on the pharmacy aspect, but we are going into some other aspect, like after completing B pharmacy and M pharmacy. So we are going into the jobs related to computer, medical coding, other things. But the core, what is the core is the R&D. So we are diverting ourselves from that R&D part. So my request to all the students that there is a lot of scope, and there is a lot of scope in the pharmaceutical industry to grow a lot in the worldwide. So if you focus majorly on the research part, okay, if you after completing B pharmacy and M pharmacy, if your only focus is that how to get into a research. So I'm telling you that is a very core part where definitely after 5 years and 10 years, you can see yourself at a very high level position. Instead of that, diverting yourself that some other areas like the computer based jobs or some other medical coding or other based jobs. So my request to all the students, so take the opportunity of this kind of conferences conducted by JNTUH frequently, and keep in mind that the only motto should be that you should get into a R&D side. That is the only request to all of you. And once again, congratulate all the students, those who are be a part of this conference. And thank you once again for giving me this opportunity. Thank you. Thank you.
Uh, Hello everyone, my name is Aditya. I'm from Malaradi College of Pharmacy, pursuing my PharmD first year. I'd first like to thank my college and APR and JND for conducting such a informative event. And I've learned a lot of drug delivery systems. Thank you for giving me this opportunity.
Anybody else want to speak? Anything? Girls? Any girl? Okay. Looks like they are not. Um, come back in a minute.
So for exam, I was present, sir. Not to write sentences.
Yeah, and put closer table. And it is so frustrating. When the COVID came, try to
Okay, participants, our program, half an hour session, and first to your respective places where you need to present it. Okay, okay. Amma, are you giving me the slide changer? It's on each side.
हाँ मैम 31st इंटरनेशनल कॉन्फ्रेंस is going on. International conference.
Savani, you put everyone.
So they are
Please.
started
What's going on?
Yes, I'm muting everybody.
I will start the session. We'll take
Yes,
for
ma'am. Yes, you can please go ahead.
and then
Unmute us.
yeah. भैया मेरा slide वो दिखाओ 4 point. Okay, full screen. Okay, I'm being the pharmacist. I want to talk about little about the career opportunities. Career opportunities of pharma field here and abroad. Okay, this is my small presentation. Okay. After completion of like 4 years of B Pharmacy or after completion of Pharm D of 6 years, you will be called as a pharmacist. Pharmacist word has 10 words, 10 letters. Each letter has its own importance because P like, P for functionality and H for humanity, A for acceptability, R for reliability, and M for morality, and A for ability, cordiality, and integrity, sincerity, and tolerance. I always feel proud to be a pharmacist. To feel proud to be a pharmacist, we should have all these qualities. Okay? And we cannot have 9 out of 10 or 8 out of 10. We must have 10 out of 10 to become a pharmacist and give a service in pharmacy. So what is actual responsibilities of pharmacy or pharmacist? First thing is you are a companion to doctor and you are a counselor to the patient. And you are educator to the society. You are custodian of medicine. People always think that after completion of Pharm D, we have studied 6 years of Pharm D and we are equal to doctor and we can write a prescription, we can do practice and all. No, you cannot do. You are not licensed to practice or write any prescription. You are only licensed to companion with a doctor, dispense the medication, counsel the medication, educate the society, and you are, most importantly, you are a guide and companion to doctor. Okay. And many people like me also, when I completed my master's, I wanted to become a pharmacist in US because, US, a pharmacist is equal to doctor, and many prescriptions are given as suggestions to the doctor to prescribe by seeing the diagnosis of a patient. In order to become a doctor, in order to become a pharmacist in US, you have a lot of things need to be done. First thing, the credits has to be equal. Then you need to pass the foreign pharmacy graduate equivalency exam. Then you need to pass the test in spoken English, and you need to pass the TOEFL, and you need to pass the 1500 hours of internship. Then you need to write the exam of NAPLEX or a California board or Florida board. Then you become a licensed pharmacist in USA. Once you become licensed pharmacist in USA, you don't have to look back anything. You will have a very good pay package. You have to work only 40 hours per week. If you work 1 hour extra also, that has to be paid 1.5 times. So you will be making the $500,000 per year. And if you want to work, the NABP requirements are that Arani, can you change the slide? Next slide. Next slide. Okay, you want to become a pharmacist in Canada. It is one of the best country that you can work as a pharmacist. And even in the Canada also, your credentials will be evaluated, and you have to write 2 exams. One is called PEBC1 and PEBC2. And you need to pass the IELTS. IELTS is nothing but a English language testing services. That is minimum requirement. Then once you pass the 2 parts qualifying exam, the theory and clinical, that is mainly based on the MCQs, and you have to do about internship for 6 months. Then you can work as a registered pharmacist in Canada. Next slide, please. And if you want to work in Australia, another one, a very good country, kangaroo country, and you become pharmacist in Australia, you must complete a tertiary degree in pharmacy, such as like bachelor's or master's program. And you need to apply for the Pharmacy Board of Australia to complete the internship, approximately 1 year. And you may need to pass the examinations like Graduate Australia Medical Examination Test and Medical College Admission Test. Once you clear and do the internship and credits are evaluated, you can work as an Australian pharmacist. Next slide. And you want to work in a United Kingdom, it is very close to us. You can just travel 12 hours by flight. And your credentials, your degree is accepted in United Kingdom, UK, but you need to pass the exam, the Overseas Pharmacist Assessment Program. Poster. Okay. Okay. So it's an Overseas Pharmacist Assessment Program. Will be assessing the your credits, and you need to write 1 exam. That is a UK pre-registration exam, and that after clearing that, you have to go for the internship of 1500 hours. Then you can write this exam. Once you pass, you can work as a registered pharmacist in UK. Next slide. Registered pharmacist in Germany. This is the best country, and your credentials are equal to the German pharmacist. Only disadvantage is that you have to learn Germany. That is a prerequisite to work in Germany. And your exam will, your credentials will be evaluated, and you have to write, educational qualification exam, that is called Federal Pharmacist Regulated Exam. And once you pass that one, your credentials are evaluated, then you can work directly. But German language, you have to get it in minimum score. That is a must. You don't have to write the English exam, but Germany exam has to be written. Next slide. You want to work in New Zealand. This is another good country that you can work, and you always, they will be evaluated your credentials, and you need to pass the English language exam. Then their CAPS exam. That CAPS is Examination Knowledge Assessment of Pharmaceutical Sciences exam. That exam is prerequisite to work in New Zealand. Next slide. Next slide. And what are these IELTS? IELTS are nothing but a international English language testing system. This international English language testing is nothing but they will be checking your communication skills, because you want to work as a pharmacist, you need to communicate to the patient and also to the doctor. So these IELTS are important for any other country other than United States. United States, you have to pass the test in spoken English. Okay, and also you have to pass the TOEFL exam. That is a prerequisite in United States, but any other country, Germany or UK or New Zealand, or you can, any other country you can take, Australia, you need to pass the IELTS. So I request all pharmacy students, beside just studying the B Pharmacy or Pharm D, you need to improve your communication skills, because IELTS exam will be conducted in listening, reading, writing, and speaking. Because we can do the writing, listening, and all, but speaking is going to be very difficult, because you have to talk on the microphone the question which was asked by them. Like they can ask you to explain a favorite actor, or explain the favorite administrator, or a politician, or a famous monument, or famous city. Any question they can ask, but you need to speak without grammatical mistakes, without any any unnecessary words, like up to 5 minutes. So that is the most important one. Many of Indians will fail in speaking skills. So I request all of you, try to learn, try to communicate, try to, English is the one of the worldwide acceptable language. Try to learn in that. Next slide. So what is your career planning? That is more important, because you have to be thinking about your career planning once you are in a first or second year. Want to work in a production department, you want to work in quality control, you want to work in clinical pharmacist, you want to work in regulatory, you want to become a pharmacist, you want to be an administrator. That all need to be think before you complete your any degree. Because after completion, you have to mold yourself to work towards to that work, that career. Okay, next slide. So I go very brief. What are the opportunities in India? Like you can work as a research development after completing B.Pharm, M.Pharm. Next slide. You can work as in a clinical research. You can have several opportunities in clinical research. Next slide. This is also continuation of clinical research. Several prospects are there. Next. You can work as a contract research organizations. The contract research organizations, they mainly do a lot of clinical oriented works, or clinical report work, or medical writing works. There you can work as a contract research organization. Next slide. And you can work in regulatory affairs. Now the boom is in regulatory affairs, all total Hyderabad. Everybody is asking whether you have a student of regulatory affairs. I don't know how this boom has got it, but it is a high boom is right now. So once you learn in regulatory aspects, now it is the pay is also very, very high. Next slide. Next slide. Next slide. And you can work in a sales and marketing. Sales and marketing, whenever you want to go, if you are interested, only go to that. Okay? Because sales and marketing require a lot of talking, lot of convincing to the other people to buy your products, and you need to have a very good communication skills. Next slide. You can work in product management and medical marketing. Product management and medical marketing is more equipped to the R&D and market research benchmarking. This is also required a lot of skills, and you need to know that complete knowledge of the topic where you are working. You cannot work this area half with a half knowledge. Okay, next. And medical transcription, medical writing. This is a most PharmD students are doing right now, because this is one of the hottest topic now, because several other country people, they just record and send you the audio. You have to listen and write your medical reporting about whatever they are described at the patient or hospital or medication or diagnosis or anything. So this also required a very good communication and English language. Next. And you can work in advertising. You know, design the advertisements, you can speak, you can act also. Next slide, please. You can work in consultancy, like strategy and planning of any company, or strategy of planning of any innovators, strategy of planning of any startups also. Next. And you can work in a business development, international business development organizations. This is the most important ones now, because many startups have come. I don't know whether you have visited any startups in Hyderabad. There is a big startups in University of Hyderabad that is called BioNest. There are 40 pharmaceutical companies started by young generation. They started within one single room of their dream. They wanted to become a employer, not the employee. So that kind of startups you can go for and work and do the business development for on your own. And Telangana government supports it, any business plan, and you can present it, the seed money also given. Next. International marketing, international business. This is also export and import business. And you know that 90% of antiretroviral drugs are supplied by India, and about 70 to 80% of vaccines are supplied by India. And almost 100% of generics are supplied by India. So you can better opportunity of all pharma graduates in this. Next. Okay, next. Next. Next, same thing, the opportunity. Next one, it is a quality control, quality assurance. These are all decided. You work, many of them analysis background, chemistry background, they go for this. And every company required this one, quality assurance and quality audit, and also quality control. So you can work in different different job roles in quality control and quality assurance. Next. Next. Medical reviewer and advisor is another one. Medical reviewer and advisor. And also this is another good job. And supply chain management is also one of the important roles. Next one. And patent agent. You know that each patent, if you get a patent and publish, then you can have a rights up to 20 years. So on that patent. So you are going to be one of the richest person that once you get the patent is published. So you can work as a patent agent for any kind of innovative product. Next. There was a talk for several years. There was no job opportunities for PharmD. But you can see here, first I thought I will just put couple of them, but I came to know many job opportunities, and there is no place to put their opportunities on my slide. So there are several opportunities, but we need to get ready to grab them. Next slide. Also same thing. So many positions for PharmD. Next. Okay, next slide. Okay, can you see this slide? What is happening here? The person whoever driving, he has miscalculated the height and he went on a bridge, under the bridge, but he got stuck. Why I'm taking this slide and telling you, okay, as a pharmacist, our pharmacy students don't get stuck anywhere. You make your career path. Think twice, 3 times, 4 times, but dream big and never get stuck and try to pass through that and never do any mistakes like this in your life that will haunt you total your life. So don't cheat anyone, don't cheat any customer, don't cheat any doctor, don't cheat anyone, but make your career straightforward and work for it, dream big. Next slide. Can you see this slide? What is happened here? You guys can see, isn't it? No? Nobody's saying yes or no. What is happening here? The driver is very reckless. He doesn't even have a patience until he fills a petrol into his tank. So he drove. So the petrol pump is also came with him. Why I'm put this slide? In your career, you cannot take any decision right away. You cannot judge any patient or any doctor right away. You need to think. You need to evaluate. And you need to confirm before you talk. Okay? Don't take any vast decisions instantaneously. Okay? Try to think, try to make your career in a brighter way. Next slide. What is this? You see a lot of people like this. They are very reckless in doing job. चले हो गया हम कर दिए बोले हम कर दिया बस. But whether we have done in 100% correct or not, that is more important. Not doing the work is not important. Doing the work 100% is more important. Okay, 100% correct is more important. Don't do like this. Don't go for the side businesses. Don't go for the chalega attitude in your life. Next slide. So you are contributor for all these things. Okay, next slide. There are so many errors happens with a pharmacist. This is a statistics. Next slide. And these are the areas we can have a medication errors. Be careful. Next slide. And there is a several errors will come. We are the responsible for those errors. Okay, we have to be very, very careful. Next slide. Next slide. Next slide. Next. Examples. These are just medication error examples. Next slide. Next slide. Next slide. Next. 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 So what is this? They look same. The bottle looks same. And they start with the same C. There is a possibility of mistake is done by the pharmacist. Instead of ciprofloxacin, there is a possibility of taking clarithromycin. So don't even trust your eyes. Check with your eyes twice before you dispensing correct medication to any patient. Next slide. Next slide. See, there are 3 different medication with the same name. So never overlook. Never overlook. Never trust your eyes. Never trust anyone. But you trust yourself and check twice, thrice before dispensing or giving the medication. Okay, next slide. Okay, this story you can read in online. It's one of the best story, and that is a wake-up call for all pharmacists. Next slide. Emily Jerry story. Next slide. Okay. Okay. This I'm not going to talk. Adi close chesandi. Dr. Hoser video ఒకటి play చేయండి. అది క్లోజ్ చేసి అదే దాంట్లో యా that one in a full screen. Voice రాట్లే. ఓకే, అదర్వైజ్ మైక్ పెట్టండి అక్కడ. అక్కడ ప్లే అవుతుందా? Okay, otherwise play it. I'll tell them. Just watch the video, then I'll discuss, okay? Yeah, go ahead and play it. అన్ని క్లోజ్ చేసేసి అదొక్కటే ఓపెన్ చేయండి. క్లోజ్ ఎవ్రీ విండో, క్లోజ్ ద విండోస్. యా, స్టార్టింగ్ ఫ్రమ్, ప్లే ఫ్రమ్ ద స్టార్టింగ్. యా, జస్ట్ look at that. What happened there? Can anybody explain? What happened? There was a patient came with a asthma. There was a MDI given for asthma, and she went home and came back and said that your given asthma medication is not working. She told to the doctor. Then he said, "Oh my God, how come it is?" Then he asked me, "Show me your MDI, the meter dose inhaler, how it works." Then she said that I am not an idiot like you. You are given the medication which is not useful to me. But then he asked, yeah, I might be an idiot, but can you please show me how you are using? How she is using? She is using like a perfume. She's supposed to put her MDI inside the mouth and take puffs. That way it will be relieve the asthma. Who is the mistake at this point? No, not the patient. Pharmacist is the mistake. Pharmacist is the last person given the medication to her. And they have to counsel the patient how to take the medication, how to use the medication, how to do the medication. Okay? So we have a complete responsibility of every sick and customer patient on us. So don't, please take care of our profession in a professional manner. And I request you all, do your best and become a proud pharmacist in your life. Probably remember these words once you become a good pharmacist. Okay? And thank you. I can take a whole day also classes because I am like that, but I will end this one. And you stay and you go to the respective presentations. We'll start the presentations until 1:15. Then you can have a lunch there at the very same area where you have and complete your presentations. Then around 4 o'clock, we'll be announcing the best poster, best oral presentations, and we are going to give the certificates for that also. And we'll conclude everything by 5 o'clock, okay? Or 4:30 if possible, okay? And thank you for attending this course. And in future also, if you get a chance, you can come and enjoy our hospitality. Thank you.
Can we get another mic?
और एक mic.
కొంచెం దగ్గర పెట్టుకొని మాట్లాడాలి
Sir. Student getting pandemic speak. Seems to be on the so
ఒక్కరికే సర్టిఫికేట్ కలుసుకోవాలి ఇక్కడ బయట హెల్ప్ చేస్తున్నారు ఇక్కడ కలెక్ట్ చేసుకోవాలి చెప్పి దాని ఇక్కడ
इधर बैठे मैं बैठी कहीं बैठी
ma'am, we're ready for ma'am.
మ్యామ్ కానీ
Only 5 minutes. But they mentioned 10 minutes, like 10 minutes lecture kadanడి. Oral. కొంచెం ఫాస్ట్ గా అవగొట్టేస్తాను. చెప్పండి నీ షాప్ చేసాను షివరింగ్ వచ్చేస్తున్నా మాట్లాడడానికి
No. Sign this.
क्या कल्पना
ये कल्पना ना
Good afternoon, everyone. It's me, K. Kalpana, from Aditya College of Pharmacy, affiliated to JNTU Kakinada. I'm presenting with my friend.
Good afternoon, everyone. I'm Somesh, student of final year B Pharmacy coming from Aditya College of Pharmacy, affiliated to JNTU Kakinada. So, technology. We know technology is using worldwide in a very great manner. So, studying pharmacy course, being pharmacist, where we are using and how can we use technology and how can we advance the technology in pharma industry. So, which was the topic title entitled as Real World Data in Pharma Industry.
What is real world data and what is real world evidence? Real world data is a data relating to patient healthcare status and delivering healthcare routine from variety of sources, which helps in pharmaceutical development and drug development.
So real world evidence, it is the evidence that resulted from real world data where we analyze any medical product usage or benefits or its potential risks. When you analyze this data, we'll get the real world evidence, which can be helpful in using technology in pharma industries.
Why it is needed? Why real world data and real world evidence is needed? Real world data and real world evidence are valuable across entire pharmaceutical companies, where it helps in pharmaceutical product life cycle, examine patient outcomes, to supporting disease education and awareness, measuring potential, measuring measuring patient adherence, and monitoring safety and efficacy of product and beyond.
Once we get the real world evidence from this real world data, so we can ensure that its effectiveness and safety data. Now, pharmaceutical industry is essentially depending upon Thank you.
Are they following industry that such type of
Yes, ma'am. Yes, ma'am. They're using.
Like in drug development and regulatory approval decisions, they are using this real
As I
world
mentioned previously about the benchmarking survey, ma'am, they conclude that 90% of people are using and some people are planning to use this real world data. And there are already some drugs which are approved by the FDA basing upon this real world evidence. Actually, we didn't study more about this.
future aspect? About the future challenges.
Actually, coming about future challenges, this real world data is helpful, but same time we are also having some challenges like where even if we are going for any human on a chip, we are going any advanced, but never human resemble in any other structure, ma'am. So it would be, for example, human body is entirely different. Even we are comparing with any chip, but still we are having some challenges in that, ma'am.
Studies
Actually, not about studies, ma'am. The industries are using electronic medical records from hospitals, from any industries, from surveys like that. They're using electronic medical records. Though on the basis of the record reports, they are concluding this real world data and analyzing, and they're getting the real world evidence from various and from directly patients also. They provided some app, My Studies app, where the patient can directly register and what we call it can directly impose their medication symptoms and all. Thank you.
Thank you, ma'am.
Thank you all.
Ma'am, Akshay Reddy, ma'am. 170.
Akshaya,
Yes, ma'am.
Akshaya Reddy, okay.
Without poster.
Yeah,
without
you can
the
explain
poster,
in
you
a
can
own
explain
way.
it.
You are going to present poster presentation. Tell me what are the objectives, what is the principle? Just briefly you can explain.
Good afternoon, respected international conference audience. I am Akshay Reddy, pursuing B Pharmacy first year at Mallareddy College of Pharmacy. So today, my aim of my poster is to reveal the most interesting and rapidly developing field of pharmaceutical nanotechnology. So with the power of nanoparticles, scientists are now able to deliver the drugs more efficiently and effectively. More efficiently and effectively. Can you please zoom it? Yeah. So our main topics are here: drug delivery systems, improved solubility and stability, targeted drug technologies, and reduced side effects. So with improved solubility and stability, it can dissolve more efficiently and effectively, which increases the surface area, which increases the surface. With its very less size, it can release its medication very. Actually, drug delivery system, targeted drug delivery, and the reduced side effects.
What is targeted?
Targeted to specific cells or tissues, we are sending the medication through. It can absorb more. It can absorb more efficient, ma'am. The bioavailability and everything.
There's the advantage in nano medicine. Future advantages and technology. Okay.
Sign here. Sir, oral poster check this.
One more.
Both are
Deccan School of from Deccan School of Arts.
Year 19. Fatima.
It's not here.
Not here.
Not
Without.
You're from day
enrollment.
one. No, day two. Day two. No, name is not here. Sir, the name is
Name not.
Day two presentation, sir.
Day 2 presentation, yes. College name?
Deccan School of Pharmacy.
Once you meet there, registration test.
Okay, then
Next. Huh?
wait.
In that, you
that-
can find ask
You
the
can ask
registration.
Registration test.
Okay.
Okay. Next.
Yes,
Ready now? Presentation ఎవరైతే ఉన్నారో ముందుకు వచ్చేయండి. వాళ్ళు వస్తున్నా కొద్దిగా పెట్టేస్తున్నా ఏదో ఒకటి పెట్టుకుంటాను. మీరు రాసుకోండి మీ పేరు రాసుకోండి. మీరే రాసుకోండి.
KVK college of yeah.
Good afternoon, one and all. Respected all the dignitaries who have presented here. My name is Sakshita and I am pursuing my B.Pharm third year in KVK College of the Pharmacy. Here my topic is the formulation and the development of gastroretentive floating tablets of cephalosporin. Here in this, I designed and estimated the cephalosporin tablet to encompass the gastric residency time and to increase the bioavailability of the tablet and to also increase the absorption. In this, my introduction, I want to tell you that as you all know that the oral route of administration is the most convenient route. But coming over of the years, but this route has been sophisticated. So here I am using the CRDDS delivery system. That means controlled release drug delivery system. In this system, the drug is released controlled and the constant of the drug is reaches to the plasma and all the tissues in the constant level, and it has the more patient acceptability nowadays. Here I choose the proceeding for this formulation because it is an antibiotic and it belongs to the third generation cephalosporin, and it is a prodrug. And in this, the TF of the cephalosporin is the 2 to 3 hours. So to improve its bioavailability,
And to increase the gastric resistance time, I have chosen this for this formulation. And next, coming to the materials, in this formulation, I have used the active ingredient. I also the polymer HPMC, hydroxypropyl methyl cellulose of different grades, and the sodium bicarbonate, magnesium stearate, talc. And I have used these ingredients inactive form of the active drug. Madam. First, it will be in an inactive form, and after when it's going to the inside, activate inside the. 12 compositions and formulations I did. The different excipients I have used hydroxypropyl methyl cellulose of different grades. This is a polymer, and I also use the sodium bicarbonate citrate, which has the effervescent nature. The gas producing ingredients. Formulation is considered as the best because the dissolution I found dissolution parameters. The dissolution of the drug is more than 720 hours. It means 720 minutes is approximately 12 hours. By that, I can include that in I could do all the ingredients mentioned in the US2, madam. I formulated parameters. I have studied the angle of repose, madam. Bulk density. Paddles.
मैं यही खा रहता हूँ इसके बाद
HCl 0.1.
Good afternoon, one and all. Good afternoon, one and all. Myself, Divakar from KVK College of Pharmacy, affiliated to JNTU. On conference, my title is Design, Formulation and Development of Sustained Release Tablets of Cefixime Trihydrate. The basic rationale of Cefixime Trihydrate tablets is to optimize biopharmaceutical, pharmacokinetic and pharmacodynamic properties, and also sustained release oral dosage forms are demonstrated to improve therapeutic efficacy of the drug by prolonging its by maintaining steady state concentration in the plasma. Need for study. The basic need for development of sustained release tablets is to prolong the half-life of the drug, which further improves patient compliance by reducing the frequency of drug administration, minimizing the local and systemic side effects. Cefixime trihydrate tablets was prepared by using polymers like hydroxypropyl methyl cellulose because it has a greater flexibility to release desirable drug release. Cefixime trihydrate sustained release tablets. It is a cephalosporin drug, third generation cephalosporin drug. The different formulations of Cefixime trihydrate was prepared by HPMC, hydroxypropyl methyl cellulose is used as a polymer, and magnesium stearate is used as a lubricant, microcrystalline cellulose as a diluent, and talc as a glidant. Evaluation parameters are friability, hardness, next, and average weight of the tablet. Average weight is calculated by initially taking 20 tablets and weighing them on an electronic balance individually. 20 tablets. Okay, thank you.
Shampari. Good afternoon to one and all. Myself Shyam Parikh. I am from KVK College of Pharmacy, B.Pharm third year student. And today I am going to present my poster presentation on cholangiocarcinoma, which is also commonly known as bile duct cancer. And it is caused due to the it is a common bile duct cancer, and bile duct means it carries biles from from liver to gallbladder, and from gallbladder to small intestine. And bile means which digests fats which we eat, what food we eat from the. And coming to its etiology, and the cause is still unknown, and main cause or unknown cause is such as cholangiocarcinoma develops from the tumor cells, and it proliferates the cells to multiply rapidly, and it forms mass of cells or tumor, which causes cholangiocarcinoma. Signs and symptoms of this disease is jaundice, abdomen pain, significant weight loss, and significant weight loss. Cholangiocarcinoma. No, it's a disease, cancer. Cholangiocarcinoma. Cancer of bile duct.
How many factors can influence?
Factors such as what we call like factors such as
The main statistics in the population.
it is a very rare disease. It is very rare disease. It's caused 3% of GIT cancer. And it is nearly about nearly about 100,000 cases in India, active cases. Yes, ma'am. Cisplatin and gemitasib. Cisplatin.
Plastic. Plastic.
Yeah, and its combination with gemitabin.
Good afternoon, one and all. The respected lecturers who are present and my dear friends. Today, my topic is euphosomes. It is a advanced drug delivery system. We all know we have been seen liposomes, neosomes, etc. are the drug delivery system which have been gone right now. But right now we are seeing the euphosomes. It is a targeted delivery system. It is made up of unsaturated fatty acids. It is fatty acids are fatty acids are known as two types of fatty acids. Linoleic and oleic fatty acids have been used as a fatty acids. In phospholipids and other neosomes, we use it lipids like phospholipids. They have for synthesizing, it is very easy, but naturally we can't find it. But fatty acids are found in body, so it can be enter into the skin very fast. As we all know that skin is the physical barrier but the drug which has entered into the body through fatty acid, it can enter very fast and it can be entered into the target delivery system. And my excipients like chloroform, I have been used it, and it has 3 methods: thin layer, thin film hydration layer, where we use the chloroform as a solvent, and we can add the excipient linoleic and oleic acid, and we can form hydration by evaporating the chloroform totally. And we are using the hydrating medium. We can form the film layer, and we can use by addition of alcohol also is the another method. And my excipient key features which have been used is are fatty acids and also cholesterol, where fatty acids will maintain the membrane, and also cholesterol will be closing the particle which where there is a euphosomes. Euphosomes is drug delivery system, ma'am. Like when we have been forming a drug, directly we are entering it into the skin, ma'am. But euphosomes are fatty acids, ma'am. So drug will be inside and it will be coated with the fatty acids, ma'am. So as our skin is fatty, we have both layers, ma'am. So when the drug entering into the fatty acids, it can pass very faster. And after entering into the particular area, it can open, it can form structure again, ma'am. It has a long sustained activity.
It's okay. Can you tell the marketed formulation available under?
I will, euphosomes, ma'am. No, ma'am, I have not been searched the formulation, ma'am, but I know that we can use it in cancer cells and also inflammatory activities. And also other also, ma'am, like in fungal also, ma'am, like fungal, it has been in fungal diseases, there is no perfect tablet, no, ma'am. It will be reoccurring. But when we are using this, it is very prolonging, no, ma'am. So for fungal, it can be used.
Such a current formulation available in the market.
No, ma'am, it's available, and it is been again, it is advanced, ma'am. There is researches are going on, ma'am. There is differences in pH and buffers. Continuous right now, research also is going on. Thank you.
Good afternoon, respected dignitaries and my dear friends. Today, my topic is about formulation and development of Today, our topic is formulation and development of fast dissolving tablet known as zolmitriphan. Zolmitriphan is a fast dissolving tablet, which is it comes to the category of the drug is anti-migraine drug. So let's know what is migraine. Migraine is a primary headache disorder which lasts for few seconds, for few hours, with or without aura. Aura is a perceptual disturbance. By some people with epilepsy or migraine. The present studies are aimed to produce the fast dissolving tablets, the drugs which act on the serotonin selective receptors, and it also comes under the category of selective serotonin receptor of 1B and 1D subtypes. These the drugs which acts on the anti-migraines are known as triptans. These are prepared by the direct compression method. Materials used such as microcrystalline cellulose, magnesium stearate, mannitol, talc, cross caramellose sodium, cross povidone, etc. Tablets are prepared by the direct compression method. Coming to its pre-formulation studies and evaluation studies were evaluated like weight variation, hardness, friabilities. It is Title fast dissolving tablets, Zolmitriptan. Fast dissolving tablet. Which drug? Zolmitriptan. Mitriptan. What is its category? Anti-migraine. Migraine, when it acts on the serotonin receptors, it activates the 5-HT1B and 5-HT1B receptors, and it inhibits the action against the vascular brain.
How many formulations you have done? Eight formulations. Which one is good?
At sodium starch glycolate. Formulation 7, ma'am.
What are the main components in that?
7 formulations. So excipient ratio was 1:0.25.
First year, second year, and year we What is conclusion? What are the conclusion?
Conclusion, we observed at formulation 7, the disintegrating time and the dissolution time was at 7 minutes and 33 seconds. Then lambda max was measured at UV visible spectroscopy at 227 nanometers.
What is dissolution?
Dissolution, the drug where it dissolute at a particular time in a dissolution medium. It completely dissolute.
What are the USP dissolution conditions?
USP dissolution conditions.
It's okay.
Thank you, ma'am.
ఇది అయిపోయినాక లంచ్ వెళ్ళాము మళ్ళీ 2 ఎంతమంది అయిపోయింది లంచ్?
7.
12 members ఉంటారు. సరే మేడం. ఇతను అయిపోగానే లంచ్కి వెళ్ళాం. Name.
Very good afternoon, everyone. I'm Tarun and I'm pursuing my B.Pharmacy second year from Geetham University. Before I start my presentation, I would thank all the people here for giving me an opportunity, and I would also thank my mentor, Haima ma'am. Coming to my presentation, my topic is about oral disintegrating tablets. This we can also call as fast dissolving tablets and mouth dissolving tablets. The ODTs are those tablets which are uncoated, and we keep it in our mouth, and it will disintegrate or disperse within 2 to 3 minutes with the help of the saliva. As we all know that we use vitamin C tablets. This is one of the good example for the ODTs, which we use during the COVID times. As a recent survey, it is estimated that half of the population we got around people having problems about swallowing the tablets. So in this, ODTs plays a very crucial role in it, and are people suffering such as psychotic diseases and geriatric patients, pediatric patients, and bedridden patients, it would be really helpful of ODTs and all. And coming to the advantages, it gives it doesn't give any ODTs, oral disintegrating tablets. Tablets. Such as vitamin C, which we use, ma'am. And there are the main ingredients we use in the ODTs are amphetamine, ma'am. As I can say that many of the people have a problem of swallowing a normal tablets, ma'am, capsules, because like small children to older people, because of some many reasons. And these tablets help them. Without intaking this tablet, we do not need any water to intake these tablets. And yeah, ma'am. There are two different techniques. One is conventional technologies and patented technologies. Conventional technologies include freeze drying, molding, sublimation, uh, spray drying, dispersible technology, cotton candy techniques, and etc. Ma'am. And coming to patented technologies, uh, ma'am, from the past, this ODTs, they are in developing from the past decade, ma'am. So there are many technologies and techniques are coming right now. And recently, though, this Durasol technique is most useful right now. Right now, I didn't keep it in a poster. So thank you.
అందరు లంచ్కి వెళ్ళి 2 PM ఇక్కడ ఉండండి. లంచ్కి వెళ్ళండి. ఎగ్జాక్ట్లీ 2 PM రండి ఇక్కడికి. 2 o'clock స్టార్ట్ అవుతుంది.
तो मालूम होगा तभी तो अरे हाँ 15 मिनट पीछे क्यों हाँ पीछे क्यों आप लोगों से मारो नहीं हो रहा हूँ लाइव लोकेशन ना तो तुम 25 हो रहे हैं फोन तो पॉइंट आया तो 2 आप लाइव लोकेशन ना तो नहीं दिखा रहा ये दिखा रहा था का डिवाइस होगा क्या हुआ ये फोटो है ना ये फोटो बन डालो मैं क्या मालूम मैं लिया बैटरी फुल है ये नहीं हो बैटरी फुल है ना कैमरे हाँ निकाल दो फुल कैमरे की लगाओ कैमरे की मेरी की 2% है ना चार्ज करा फुल अच्छा हटाओ तुम क्या हाँ जरा
आने रहे
क्या क्या नहीं
Only harm could get found out. Maturity. I just couldn't make up. George decided he would fire
Sandeep Chaurasia. My thumb. Take that. Okay, so can I start? Can I start? Yeah, myself, Dr. Sandeep Chaurasia. Presently, I am working as a techno-commercial lead scientist in the formulation R&D department. Yeah, in at the Ashland India Private Limited is a US-based multi-specialty company. So I did my PhD from IIT BHU, and today the topic is of design, development, and optimization of stable lyophilized protein formulation by using viallose trehalose dihydrate through the freeze drying process. So next slide. So introduction of this presentation, the critical consideration to select the selection of the excipients and the manufacturing process. So generally different excipients are used in as a lyoprotectant and cryoprotectant for stabilization of protein and peptides or macromolecules. And the lyophilization steps is involved to protect the drug from the degradation process. So the the specialty or function of the cryoprotectant and lyoprotectant which are used during the lyophilization is the first of all, it acts as a stabilizing agents to stabilize the protein and peptides from the degradation. Second is the it acts as a cryoprotectant, means it protected and enhances the shelf life of the particular macromolecules. So the steps involved in the lyophilization is the freezing, first steps, then sublimation, that is a primary drying, and then secondary drying, that is a desorption. Now coming next slide. So the objective of this study is to determine the upland glass transition temperature TG prime of the TG prime of the 5% viallose trehalose dihydrated by MDSC study, and to determine develop and optimize the lyophilized protein formulation using extreme vertices mixture design using Minitab, and then identify the appearance, pH and assay, then moisture content, reconstitution time, and aggregation study of the bovine serum albumin of the lyophilized formulation. Next slide. So these are the material and methods. So these are the total 17 run candidates are now run through the Minitab software, and out of 17 we optimize the formulation. Out of 1 give the good composition with respect to their moisture content, time, reconstitution time, and pH, assay, as well as the aggregation study. The lyo cycle means a lyo recipe is of 44 hours 35 minutes. Next slide, please. So this is the TG prime of the 5% trehalose solution. So heat cool heat cycle is performed to identify the TG prime, that is up to -28 degrees centigrade is observed. Next slide. So this is the appearance, pH and assay. So the most of the formulation is showing the intact cake, but few are showing the shrinkage and cracks during the lyophilization. This is the defect of the lyophilized formulation. Next, please. So in this the pH is in the range of 5 to 6. The standard acceptable range is the 4 to 8, and then assay is in between the 95 to 110%. Next is the moisture content. With the help of DOE, we found that the moisture content is in the range of 3 to 5%, but the optimized formulations F15 is the optimized formulation is showing the moisture content is less than 3%. Next. Reconstitution time, it is also is in the range of 21 to 52 seconds, but this is one of the video at the corner of the bottom, and but the optimized formulation is showing 21 second is the reconstitution time. Next. Aggregation studies. Aggregation is the of the bovine serum albumin. So is in the combination of histidine hydrochloride, and polysorbate. These are the composition of the entire formulation. So the impact of histidine hydrochloride is acts as a buffering agent, but onto the aggregation study during the heating or means kept at a 3 days at 100 degrees centigrade, there is no any aggregation is found. When it is the BSA is with the histidine means buffering system. So the histidine hydrochloride plus trehalose play an important role to protect it from the aggregation. So these are the entire. Now coming to next slide. So the summary and conclusion. So we found that the optimized formulation F15 is the optimized formulation, and their moisture content is in the range of 3 to 5%. Reconstitution time is the 21 to 52 seconds. At the bottom, these are the means of composition, means which is optimized. These are the acknowledgement and references. So any questions from the slides is most welcome. Yeah. Slide number 2 or 3. Yeah. Going to up. Up. Next. Third slide. Sorry, fourth one. This is the fourth one. Fourth one. That one. Yes. Can you keep in a slide mode? Yeah. So this is the composition at the corner here. Technique is the simple mixing method we are applied to dissolve all the components. First is the bovine serum albumin. Second is the buffering system, histidine and histidine hydrochloride to maintain the pH. Third one is the polysorbate 20. And last one is the trialol, which acts as a cryoprotectant and lyoprotectant for the particular protein.
Then finally
Freeze the product. So now coming to next slide. So next slide, please. Next.
Where are you going to use this formulation?
In our lab. Means you can we can use in a different means these are the injectable formulation. Okay. So this the final dosage form is in the injection. So when during the injection you can reconstitute with the help of saline water or dextrose solution. Sorry. API is the model protein we are using here. No, you can use any other biological molecule like peptides. This is the model, yes. But you can use the peptides, any peptides or enzymes or any biological molecules, for example, monoclonal antibodies or recombinant DNA technology based.
Like as a drug carrier.
Correct. No, not a carrier. It is a protect it from the denaturation because all the biological molecules are very highly highly means you can say that it is unstable at the room temperature or degraded very fast. Means there are so many degradation process takes place. So how it will be protect and increase the shelf life. The tree halo is having a means a specialty to protect it from the degradation and enhances the shelf life of the particular molecule. No, I am just added now this model protein bovine serum albumin. Bovine serum albumin is added here as a model. Yeah, it is stable. Yes. Yeah, thank you. Okay. Second slide. Sorry, just a minute. Actually, one slide is also.
You just wait, sir. Sandeep, sir.
Yeah.
We'll give the certificate.
Yeah, sure. Okay.
Will it certification
Go.
That will not be visible, by the way.
Sandeep,
Off. Perfect, perfect. Thank you. Thank you. Yeah. No, if you can switch off one more, I think that was better. Earlier was better. If you can. Off ही कर दो, off ही कर दो. Yes, perfect, perfect. That's okay. Even smaller lights. Yeah, perfect. This is perfect. This is perfect. Yeah. Okay. Good afternoon, everyone. Respected Chair, JNTU. My name is Anup Bankar, and I'm a PhD scholar at MIT University. I have 14 years of research experience, and I'm working with one of the MNC in Hyderabad. So this is my PhD research work. This is, I'll just be at this side. Is that okay with you? Okay, so this is development and validation of lurasidone, a drug substance, as well as drug product in the form of in situ formulation, in situ depot forming formulation, wherein we are estimating drug substance in the formulation. So next slide, please. So the outline of the research work is to develop a stability indicating HPLC method, and for DS as well as for the drug product. Next slide, please. Yeah, so what is the need of this study is currently there is no pharmacopoeial method that is available for lurasidone hydrochloride in any of the pharmacopoeia, and whatever methods are reported in the literature, that they either lack being a stability indicating method, or they have limitations of having an unsafe method, because most of the methods use perchloric acid, and that is unsafe to use in a routine quality checks. That's one of the disadvantage, and there is no formulation which is reported in the literature which can serve estimation of lurasidone in PLG or polymeric matrix system. So currently in the market there is only one dosage form which is tablet, and the currently reported methods are only directing at tablet dosage form. So there is no method as such. So this is the need of the study. This is a research gap which we are trying to fill out. Next slide, please. So this is the first slide wherein method development, spectra and chromatograms. There is a bit of a, yeah, if you can, yeah, go even more. Yeah, perfect. So this is the method wherein we are developing a new system wherein the solvent system is orthophosphoric acid 0.1% with ACN, which is 50:50. And this is giving us the spectra. The best part of the spectra in the reported literature method, the methods are reporting 8 or 9 minutes of illusion time, whereas here it is just 4 minutes. So it is a double fold of advantage to the existing methods which are reported. And the parameters for the HPLC, RP HPLC methods are already mentioned there. It is 230 nanometer wherein the spectra was found. And the flow rate is 1 ml per minute, and the column temperature is 30 degrees Celsius. Next slide, please. Yeah. So this is what is failure looks like. So this is what this is where we failed. So we tried all these different columns, all the mobile phase, all the temperatures, everything, and we disastrously failed. However, at the last part wherein we used discovery column, so baseline was stable and method was successful. This is what we adopted. In the earlier method, which we have reported, there was a failure with respect to poor chromatography, high retention times, and non-stabilized baseline in the HPLC method. Next slide, please. So system suitability and linearity was checked. So system suitability was performed as per ICH guidelines. So 6 standard injections were injected to the HPLC, and the outcome was less than 2% of RSD, which also can be seen in the spectra there. And in the linearity, we have checked the linearity from 25 to 150 microgram per ml, wherein there the linearity found was R square found was 0.9998. So it was perfectly linear method. Next, please. So yeah, repeatability and intermediate precision was performed by injecting 6 samples. It was repeatability as well as it was intermediate precision that was performed by injecting six samples. It was on the same day as well as the next day. And what we found was RSD was well within the limits of ICS guidelines. Next, please. Accuracy we have performed by 2 methods. One is by directly estimating drug substance, and also in the drug product as well. So we used a standard addition method wherein we added known concentration of the drug in the method, and we tried to estimate the concentration of the drug in the accuracy method. And what we found was it was well within the limit. We can see the mean recoveries were 99 and 100, and also the RSD was less than even 0.5. Next, please. Robustness and LOQ. So robustness for the DS, for the drug substance, was estimated for flow rate, mobile phase, and column temperature. There were different variations, those have been played. However, the method is so robust that even we make these changes, the method was rock stable. And LOD LOQ was checked and it was found to be well within the acceptable ranges. Next, please. So solution stability, we have checked solution stability for 0 hours and 24 hours. And what we found was until 24 hours, the solution can be very well used without any deterioration of the solvent systems. Next, please. Yeah, so this looks bad though. Can you please adjust? Because this is very important side. So this is a force degradation study. This is important piece of information where we have digested our drug substance as well as drug product. And this is what we found. So we did acid, alkali and trihydrate degradation, wherein acid was degraded at 1 normal HCl using 1 normal HCl at 30 minutes. And it was reflux at 60 degrees Celsius. So if you can see in the chromatogram, there are two peaks. One is a peak of interest, another is peak of impurity. So API was degraded roughly 5 to 6%. But it was very well indicated by the method. This is how it proves it is a stability indicating method. And also in the alkali, we found the degradation, but the peak of interest was very well resolved and it was very well seen. There was no degradation that was observed in the dry heat. Next slide, please. There are 3 more degradation patterns. One is oxidation degradation, wherein hydrogen peroxide was used. Next is a photo degradation, and after that there was a neutral degradation. In all of these degradation studies, our method was very well robust to discriminate between the peak of interest and peak of degraded product or impurities. Degradants per se. This is now method for drug product, wherein we have found that USP plate count was more than 2000, which is ICC requirement, by the way, and USP tailing was less than 2, which is a mandate to have a successful method, and RSD at all the cases was found less than 2, even for the drug product. Now, the interesting piece of information, it is sometimes it is easy to estimate the drug as a drug substance, but sometimes it is very difficult to estimate drug substance in the drug product. So in the matrix of polymer, So yeah, sure. So next slide, please. This is my second last slide. And there were precision that was also done. And next is, and there were also robustness studies done. Those were also perfectly fine. Next, please. Solution stability was again perfect till 24 hours. Next. And there is a conclusion. So we have been successful in developing a method which is robust, stability indicating, safe, effective, and easy to estimate, because 4 minutes was the runtime. So this was one of the successful method, and the usability of this method is this can be used in any matrix of the polymer very comfortably with the BCS class 2 drug. So this is our conclusion. Thank you for the time. It is lurasidone hydrochloride. It is antipsychotic. It is second generation antipsychotic. Yes. So not on the slides, but yes, there is a structure. So the moieties get when they get exposed to this acidic condition, the ring opens. So that's how the API gets degraded. So there is a mechanism. I also have that, but not on the slide. It's not tricyclic. It's I mean, there are only two cyclic structure, two cyclic structure. It is not tricyclic antidepressant or antipsychotic. No. Yeah.
Chemistry that is affecting
during the analysis, right? Yeah, there was a pH during this method that was playing a very crucial role. So the current pH that we have kept is at 3, because at the basic pH the evaluation was not coming well good. It was invented in summer 2017, just 5 years back. Yeah, it is new drug only. Yes, so it is a innovation. Thank you so much, ma'am. Thank you so much.
Name?
Yeah, sure. My name is Anup Bankar. Anup, ANUP, or you can
Present.
surname Bankar or BAN. KAR. AN is my name. Oh, okay, that is my surname, by the way.
Bankar,
Anup, AN.
Next, which MET?
MET.
Oral.
This is poster. This was poster. I have
Poster.
poster, but it was not visible, so I have poster as well. If you want, I can show that the poster.
Sandeep sir.
Thank you.
Madam.
Sorry. Good afternoon, ladies and gentlemen. This is Mahsa. I'm from Iran, but I'm studying in Rajiv Gandhi University, Al-Amin College in Bangalore. This is a poster I made ready for presentation, second international conference. It is about the medication error. I studied this medication. I found this medication error in San Flaminos Hospital. First slide is definition of error. All preventable even happen during prescribing the medication or labeling the medication, or during the enter the name of the medication in system as any professional or professor, they prescribe the medication, but during the enter the name of medication in system maybe happen and sorry, shall I? And according to the FDA of USA, each year above 100,000 errors happen during each year in USA. It is we can prevent the error if we will focus to the whenever we want enter or when we want dispensing the medication. First slide, type of the medication. I found in 153 cases, it was during 6 months, my research about this one. About 95 people, it's female, it was female, and 53 like this, it was male. The most error happened during the prescribing error, and after that about documentation error and administrating error. We can focus in a slide, it's very bad. Okay. About 97 case I found for prescribing error and 33 for administrating error, and around 23 was 23 about administrating error, 33 about documentation error. For prescribing error, we have problem about a route of the medication. They wrote by the mistake. For example, if it was injection, they wrote orally in front of the medical chart, they wrote orally. Like this happened. Or one case I found for drug interaction. And about 30.61% was found incorrect route. They give completely, they didn't give completely course, or maybe omit the drug, like this happened. And next one is a type of administration error. It was happened about, can you focus? Because I can't see. About 23, it was administered from out of the 153 cases, 23 cases I found about the administration error. 23 administration error. 7 administration error was wrong. It was about wrong doses. 4 administration error about wrong route, and wrong drug administration. We have like this. And can you go for again second slide? Documentation error. For documentation error, we have error about enter the lab data, about demographic, and in a medication chart, they have some error. In medication chart, because in our hospital, antibiotic, I don't know about another hospital, that's why I'm telling in our hospital, antibiotic chart is different with another medication chart, but they made a mistake. They add antibiotic in a medication chart. They didn't write especially in antibiotic chart. And in lab data also we have some error. They made a mistake by enter the number. Manually is different, systemic is different. And can you go for next compatibility? If you can focus. Okay, so I can't read, it's not readable. Second, the last one. Huh. Imputability. Shall I conclude? Okay, I conclude. We can solve this problem. It's preventing. We can solve, and it is preventable even if we will focus on work, and whenever we want to enter the medication, actually with mind we will write like this happen. Yes, I done in internship. Usual prescription error, it's about the wrong dose and wrong route of administration. Actually, I told some example. Yes, I will tell. Wrong dose. We have problem with hypothyroidism in India, or most of the world, they have same problem. For example, if they will prescribe Tylenol 50 MCQ, they will write 50 mg like the, but yes, very high dose. MCQ is too much different with MG. And first, during the definition, I define error. I told maybe it's harmful for the patient. This is one example, harmful for patient, and maybe changed his life or her life, maybe cause of die also. For about enoxaparin, if we will write wrong dose about enoxaparin, is happen for a death. Bleeding, it's happened like this. And if you have any more questions, thank you. Thank you for your hospitality. This is first time I've been in Hyderabad. You have very warm hospitality. Actually, I'm from Iran, but I'm living in Bangalore. Thanks. Thank you so much. Thanks. So good.
Write your name. Yes, ma'am.
ఫార్మసీ డిపార్ట్మెంట్ లంచ్ చేశారు కదా అక్కడేనంట ప్రెజెంటేషన్ అక్కడికి వెళ్ళి చేసేసేయండి ప్రెజెంటేషన్.
Can I start now? Very good afternoon to the dignitaries present here. My name is Asmita from Geetham School of Pharmacy, and I'm in the final year. And before I present my topic, which is solubility enhancement of rilpivirine by inclusion complex with cyclodextrins, there are two facts that I want to tell. When I went through a couple of references, I found out that approximately around 90% of the drugs that has been marketed since the year 1995 are very poor in solubility, permeability. Also, secondly, in BCS class 2 drugs, only 30% of the drugs have been marketed and around 60 to 70% of the drugs are still in processing just because their solubility and dissolution rates are very low. So that begins my presentation. Please go to the next slide. Yeah, the drug that I've taken up for today is rilpivirine. Rilpivirine belongs to BCS class 2, which has high permeability and low solubility and low dissolution rates. And it is a non-nucleotide reverse transcriptase inhibitor used for treating HIV. Now, as I said, the problem with this drug is that it is very low in solubility. So we have tried to improve the solubility by enhancing and making inclusion complex with cyclodextrins. The very two reasons why we have chosen cyclodextrins is that cyclodextrins form inclusion complexes with the drug that have low solubility, and it basically acts as the apolar part or the functional group, basically hides the hydrophobic part inside the cavity of the drug, and the hydrophobic part is exposed to the environment. So what happens by this way is that the drug has high level of solubility. So once you start dissolving in water, it immediately dissolves. So, and the second fact we have chosen cyclodextrin is because cyclodextrin is highly compatible with human body and also has and can be administered orally. So it has excellent properties. And the method that we have chosen for this project is solvent evaporation method. Next slide, please. Yeah, the aim of the project work, as I said, is to increase the dissolution and solubility of rilpivirine by solvent evaporation method. And we have also characterized by doing drug content and uniformity, FTIR, and also in vitro dissolution studies. Next slide, please. Yeah, so this is the aim and this is the method. So basically we have done 4 formulations of the ratios 1:1, 1:2, 1:3, and 1:4. Out of, by the process of solvent evaporation, basically we mixed the drug and also added methanol and also added the complex beta cyclodextrin. And also we subjected it to drying under hot air oven at 75 degrees Celsius. And the powder that was obtained was of various formulation and various ratios, out of which the 4th ratio is the optimized formulation. Next slide. Okay, so this is the standard value graph. As you can see, of all the concentrations 2, 4, 6, 8, 10, and at absorbance, various absorbance. As you can see, this is a linear graph with a calibration curve and regression coefficient of 0.9981, which means that the graph fits perfectly into the data. Next slide, please. Yeah, so here comes the in vitro dissolution studies. As I said before, we have found out that the 4th formulation is the optimized formulation. Please have a look at the graph where first one is the one of the rilpivirine, which is a pure drug, and the 4th formulation has highest dissolution rate. So once after the dissolution studies were performed, we have found out that the 4th formulation has highest dissolution rate, which means it is released at proper intervals in the body. So yeah, next slide, please. Yeah, this is the FTIR studies that we have found out. According to FTIR studies, we have found out that the optimized formulation has good drug excipient properties and there is no drug excipient reactions. So this is the FTIR of rilpivirine, cyclodextrin, and the 4th formulation that I've made. Yeah, drug content studies was also performed. So under drug content studies, 20 mg of the drug was dissolved in 100 ml volumetric flask. And yeah, okay, so drug content was also performed. And as you can see, again,
it's very evident that the fourth formulation has the highest drug content. And yeah, next one, please. Yeah, in summary, I would like to conclude that out of the 4 preparations, 1:1, 1:2, 1:3, 1:4, that was done by solvent evaporation method, fourth method has the best formulation. And also there are still a lot of methods under cyclodextrin which we are still yet to explore. So for now, solvent evaporation has been done and the results have been clearly put down. Yeah.
Good afternoon, everybody. My name is Rashmita. So I'm from Geetham University. I'm from final year. So this is my topic, formulation and in vitro evaluation of oral disintegration tablets of clopidogrel. Next. So as you know, many researchers and scientists have been working on different drug delivery systems, in which oral disintegration system is one among them. Oral disintegration system is also known as dissolving tablets and then mouth dissolving tablets, etc. So this oral disintegration tablets, basically the tablet would dissolve in less than 60 seconds. So the drug I've chosen here is clopidogrel, which comes under the classification of antiplatelet medication. It's basically an anticoagulant. The mechanism of action of the drug is when the it'll inhibit the binding of ADP and the platelet, and then it'll inhibit the activation of PG2B and then 3A, and then it'll stop the aggregation of the platelets. It comes under BCS class 2 drugs. So the main, excuse me, can you just... As it comes under BCS2 classification drug, it has low solubility and high permeability, where we have to increase the solubility. So the main objective is to improve it and then optimize the best formulation out of it. So many people, as 60% of the drugs are an oral formulation, and people like geriatrics and pediatrics and people who are bedridden can't take oral medication. They have difficulty of swallowing. So these oral disintegration tablets can be taken without using the water, so that it will disintegrate in the mouth just like less than 60 seconds. So it is used for that. So after the formulation parameters, I pasted it. Okay, I'll tell the formulation. So the formulation contents I used here is the drug clopidogrel, and then the super disintegrant, which is microcrystalline. No, it's not. Sodium cross caramelose, and then talc, magnesium stearate, and then lactose, etc. we used here. So direct compression is nothing but we mix the excipients and then the super disintegrate and the drug together without using any okay. So these are the pre-formulation parameters like angle of repose and then bulk density and tap density. Angle of repose here is used for the flow properties to test it. So as you can see, all the results, they come under the limit, under the official limits. Yeah. These are the post compression parameters, which is thickness and then friability, and then zoom karo. Hardness, friability, and weight variation, content uniformity, and the disintegration time. So out of all this 5 formulation, the best formulation is the 4th one. And these all formulation parameters come under the compliance results. Next one. Zoom, bhaiya. This is the standard calibration curve where you can see we got a linear curve, and then the R square value is 0.9599. So this is used to know that the drug will turn into amorphous, so it is soluble, and then the bioavailability is increased. And then this is the dissolution graph, where you can see the 4th formulation, the first one, disintegrated in 19 seconds. So it's the best one out of it. Bhaiya. This is the XRD results. As you can see, there are 2 graphs. The first graph is the pure drug, and the second one is the normal formulation. So the difference is, as you can see, there is a much shift of the peaks. You can say that the drug is more soluble. This is a summary and conclusion. So as we can see, all the pre-formulation and the post-formulation parameters come at the compliance results. And the 4th formulation is the best out of it. So we can say that the drug is soluble and its solubility has been increased, and it's ready to take it orally, and it will be disintegrated in less than 60 seconds. I thank Dr. Haima ma'am for guiding me, and then my college for providing me all the facilities, and then JNTU for giving the opportunity. Thank you.
Which condition it is given? Like from where exactly? Heart attacks, myocarditis. Any blood and heart. It's a blood thinner. So why do you go for this oral dissolving tablets for this particular drug? Even tablets, oral tablets are also available. Why do you want to go for this particular oral dissolve?
soluble tablets. Dissolved in a very less period of time. So people like old people and vegetarian people, they cannot take water and they won't take it. So with saliva only it will get disintegrated in less period. So what is the excipient that is helping get it dissolved? Of course. Super disintegrated. Sodium cross. Super disintegrated. Sodium cross carbonate sodium. Cross carbonate sodium. What is the use of lactose? You are telling lactose. Excipient low property. Bulking. What is bulking? What is the dose of the drug? Each tablet contains 200 mg. What the how drug is 70? See, the weight of the drug is very less. So how to handle it properly? Like will increase the quantity by using the inert ingredient lactose, glucose, something like that. So it increases the weight of the tablet. And XRB studies, what for it they are used? Not the doctors and it should be soluble. If there are typical of the beats, you can tell that.
A very good afternoon to all the dignitaries present here. I'm Tejaswini from Geetham University, pursuing my BPharm second year. Today, I'm going to present a comprehensive review on orodispersible tablets. Orodispersible tablets are also known as fast dissolving tablets or mouth dissolving tablets. They are usually used because they have good patient compatibility and they even improve bioavailability. ODTs generally they usually dissolve within a time span of 2 to 3 minutes when they are administered, and they improve the patient life compatibility like because they're easily administered. They can be usually easily administered by people with disorders and hydrophilicity excipients are usually used, and ODTs generally observe the patient's life cycle and make convenient a form of preparations for geriatric, pediatric, and psychiatric patients. The methods used for preparation of ODTs there are two types of methods: patented technologies and conventional technologies. Conventional technologies are sublimation, mass extraction, spray drying, molding, etc. And the patented technologies involve flash mob technology, nanotechnology, shear foam technology, Durasol technology, etc. And the main objective of ODT is to enhance bioavailability, to improve bioavailability, to improve patient compatibility, to improve hormone blood sugar levels. The main advantages of ODTs are they have high flavoring agents. They disintegrate very quickly when administered. And the high volume can be taken. The main disadvantages are large volume of drug is lumped at a time, and so on. The evaluation tests for ODTs are dissolution test, disintegration test, fireability, wetting time, hardness, and strengthening. The main challenge, the main marketed preparations are livostatin, diclofenac sodium, primequine phosphate, etc. The challenges for preparing ODTs are the amount of drug, the size of tablets, the uniformity of weight, etc. And coming to the conclusion, ODTs have made an easy way of administering tablets for aged people, people with some disorders. All. Thank you.
One question. What is method? Like frequently used method? A direct computation. Direct computation method is used. So what is it? So preparing coding.
Who are the participants? Come forward and check your name in the list. Who are the participants? Come forward and check your name in the list.
Because initial Ethan. Okay. Poster or oral? Oral. Third step. Very interesting.
Start. Good afternoon, one and all. I am K. Vikas from Malaradi College of Pharmacy. I am currently studying second year B Pharm. I am here to talk about organs on chips. Next. Organs on chips may be a complex term in a single word, but when broken down, it is a very simple term. Organs, as we know, is next. Organs, as we know, is a collection of cells, and chips in this case are microfluidic chips that are functional, serve as functional support and as well as mechanical support. Next. So we're talking about organs on chips. We should consider drug analysis. Drug analysis, when we manufacture a drug, if we have to take it to the clinical trials, we should affirm that it is safe. To affirm it is safe, animal testing is the most opted method, and a mouse is the most opted animal for animal testing. Next. Next. A mouse and a human has a genetic similarity of 85%, but in case of apes, it is 96%. When we show this data to an analyst, of course, he should choose apes, but in 95% of the cases, mouse is the animal chosen for animal testing. So next. There are few reasons why we choose mouse for animal testing. Next. Mouse is more convenient when compared to apes to transport or to move, and has a short gestation period to look if the drug has any effect on the baby, and it is relatively inexpensive, and it is mild tempered and cooperated than apes. Next. And moreover, apes are protected from the 2013 chimp law, and that leaves mouse as the option for most of the drug analysis. This leads most of the drug testing are done on the mouse, leaving no other alternative. But next, an organ and a chip can used as an alternative for drug analysis. This is a graph showing the red line is the amount of money spent on research and development and drug discoveries. The corresponding blue bars are the amount of new drugs approved. Next slide. As you can see, there is a decline in the rate of new drugs approved per every $1 billion spent on drug discoveries. Next. To overcome this, the WIS Institute has came forward with an idea of organs on chips. It has lung on a chip, liver on a chip, and a gut on a chip, and a few more designs on the name. Next. This is a lung on a chip. It has next black videos. This is a lung on a chip. Play video. Yeah. This is a lung on a chip. It has hollow capillaries, and the center capillary has a porous membrane in the center, separating the capillaries into two different channels. The porous membrane can be filled with real lung cells on the upper side and the capillary cells to mimic the air channel on the upper side and the blood channel in the lower side. And the vacuum channels can be used to apply pressure to mimic the contractions of a lung. And we can supply air in the upper channel to make it air channel, and the blood in the lower channel for blood channel. And this whole cell arrangement mimics that of a real lung. And we can test a few drugs on the chip, and we can see if how the drug responds to the lung. This is the liver on a chip. Like we discussed in the lung on a chip, it has a capillary, hollow capillaries as well, with a porous membrane in the center. And unlike the lung on a chip, it has hepatocytes on the upper layer. They're the functional liver cells, and endothelial cells on the lower side. Conclusion. Skip just last one. Next. చెప్తాను పైన కొంచెం ఇంకొంచెం please. So how good are my organs on chips? Next slide. Let's see with an example. These are white blood cells. White blood cells circulate all over the body, searching for any infectious bacteria or foreign cells. They can differentiate host cells and foreign cells by chemicals released by the foreign particles. As soon as they detect foreign particles, the chemicals released by the foreign particles, they slow down, they stop, they stick to the capillary walls and make their way out of the capillaries by squeezing through the cells like that. And they then follow them to the bacteria by chemical trials. Next slide. Thanks, sir. Conclusion. I conclude that organs and chip has the potential to make one of the most visionary pharmaceutical, most innovative visionary of the pharmaceutical industry, that personalized medicine can be made through by organs and chips.
Which university invented this technique?
Which institute? Huh? Which institute?
Where it is?
Germany.
Germany.
Thank you.
Sahiti Amna. Okay.
Sahiti. Sahiti. Which college are you from?
Geetam University, ma'am.
Start the presentation.
Good morning to one and all. This is Sahiti from Geetam University. My topic is about formulation and evolution of rilpivirine by using spherical crystal agglomeration technique. Next. And the three phases in this strategy are choosing the right technique to create the crystals, and then by selecting the wetting agent, which will not mix with crystallization solvent, and then the hardening of agglomerates. The main agenda of this topic is to improve the solubility and increase the bioavailability. Rilpivirine is used for treatment of HIV. It belongs to the BCS2 classification, which has low solubility and high permeability. And the work of this project is to enhance the solubility and increase the bioavailability. So next. The materials used in this formulation are urea, sodium citrate, rilpivirine, and distilled water. Urea is the binding agent, and sodium citrate is the substance which is useful in reducing the acidity. And rilpivirine is the drug which has low solubility and main drug which is used for treatment. And these are the evaluation tests which are performed: UV spectroscopic analysis, FTIR analysis, pre-formulation studies, and XRD test. And these are the formulations I have prepared. And my optimized formulation is F4. This is FTIR graph of rilpivirine. And my project is continuing, so I haven't received a graph of main formulation. This is graph of raw rilpivirine XRD test. And this is the graph of it is a formulation of rilpivirine and evaluation.
Which year you are in? B.Pharmacy?
4th year, ma'am.
The different formulations.
It is in crystalline powder form.
Why did you mention in the presentation the formulation after the evaporation? Why should you?
To enhance the solubility and increase bioavailability. The time, the presence of drug and the systemic circulation. Yes, ma'am, therefore. BCS 1, 2, 3, and 4. It belongs to BCS 2, ma'am. It has low solubility and high dispersion rate, like high permeability, sorry. Thank you. I would like to thank Haima ma'am, our college, and JNTU college for providing this opportunity. Thank you, ma'am.
Who is next?
Govardhan, first tell me how many are left over? Only name the 3 members. Gayatri Krishnan, please come for the presentation. Absent. Shyam Sundar. Shyam Sundar. Kusmita. Okay.
Presentation of the certificates. Best presentation. Morning.
She's not coming. Okay, sorry.
Presentation.
Ashwit will call
No.
Yeah. Let खोलो. Let खोलो. चलो तुम लोग कैमरा से ले लो. रोज कैमरा में ले. 1 मिनट कैमरा. ले फोटो. ये वीडियो. आ रहा है क्या? ये बोले तो.
photo. आप देते हो ना प्रोग्राम शायद जितना काम है बढ़ निकल गया ना लैपटॉप के
कैसे करते हैं ये पेन ड्राइव की ఏ అందులో కాపీ పేస్ట్ చేయరా బాబు ఏ పెట్టకుండా ఎట్లా చేస్తా సరే
Okay, good evening, everyone. So, how are you doing? Exhausted? Oh my goodness. Okay. Anybody want to come onto the stage? You can sing or you can tell a joke or anything. You want to come? No? Anyone? Just a few minutes. We are going to give you your best poster, best oral awards in few minutes. Come on, girls, boys, anyone? Or you can tell a story also. You can tell any story. No? Come, come. Or anybody want to dance also, welcome. Want to come here? You want to...
I'll use ma'am, Carnatic, I sing classical. Anything? Sing anything you can. Just a small clip, that's all. Anything you can. 1 minute. మూలాధార మూర్తి మూలాధార మూర్తి గజముఖనే చరణమునదు ఉభయ చరణం మూలాధార మూర్తి గజముఖనే చరణమునదు ఉభయ చరణం మూలాధార మూర్తి గజముఖనే வேலாயுத தோன்றிய விமலாபுமையார் தரும
மகனே அமலாயமையாள் முனிவர் தொழும் விமலா உமையாள் தருமகனே அமலாயமையாள் முனிவர் தொழும் மூலாதார மூர்த்தி கஜமுகனே. Thank you.
Okay, now the turn is from boys side. Boys side. Come on, any boy? Even twinkle, twinkle, little star is also okay. Come on, girls, boys. You should take like a challenge. Come on. Maybe boys will do dance. Yeah, come. No, not that. Okay. Okay. No? CPS girls, you want to speak anything? Okay. So anybody wants to come and give any feedback? When we do the next time, we can have some more things. Or you can express your views on this conference.
First I'll do this one.
Anyone can come. Just speak one and yeah. Give an applause to her.
Can I send you the audio? Can you play it? I send you the audio.
And next person get ready after her. Okay, I'll tell you the joke. So we all studied dispensing, isn't it? Okay. That was my first class in dispensing. And my sir was telling that we have to prepare a syrup. Okay. And that syrup will have a sugar and vehicle and some preservatives. And we have to make up to the required quantity. So what sir told us, take the sugar, weigh that one and get the vehicle and prepare the syrup. And you know the first class, we don't know what is vehicle. So I asked him, which vehicle? Is it a car? Bus? Which vehicle I have to go and get it to prepare the syrup? You understand the joke? So we were that much naive at that time. So then he has come with like 4 or 5 different bottles to the class. Said, what is this? This is water. What is this? This is oil. This is something. Then he told, these are all called vehicles. And you know that we need to require to prepare your syrup. This is a liquid vehicles. So that was a foolish thing. I mean, you know, that was very innocent questions. So probably, did you guys feel anything in the pharmacy like that? No? I felt so many. Because by asking all those things, we learn more. And we feel, you know, we will never forget those things. Am I ready now? It disappeared. Come, what happened? Not coming? Okay. So any other things? You know, whenever we joined in the pharmacy course, B pharmacy course in the, 1986, 1990, like that, 96. So they used to say, when you become a pharmacist, you have to work as a compounder. So what is a compounder? Anybody knows about it? Okay, that time we don't know that also, but we visited doctor once in a while. In the doctor place, there was a doctor room. There was a small room. In that room, one person used to sit with a red color, blue color, green color liquids. Like, paracetamol, probably. We don't know that time, penicillin or whatever. So when we asked the sir, what is the compounder job that we have to do after completion of B pharmacy? Then he took to the health center and showed that you have to do that mixing of all color liquids. Okay, color liquids. Then we thought, okay, this we can pay with a rangoli with that. Okay, but we realized that there will be some API in that until 3rd year or 4th year. We didn't even know that there was some API will be in that colored liquids, and that will have a therapeutic effect. Okay, did you guys feel anything like that? No, I think you are very advanced. Artificial intelligence. Okay, are you guys done? Yeah. What are the other stories? I can tell so many to my students, but I forget. One more story. Otherwise, I would have to wait like this. Okay. You guys want to sing? Come. Anybody can come forward and speak something. Come. Just a feedback. 1 minute feedback, or introduce yourself to the other people, other delegates. Can you define anyone artificial intelligence? Anyone? As a by pharmacy student, artificial intelligence. I'll tell you the joke, one more joke. Okay. The two, one boy and one girl. They belong to the family, same family, brother, small brother, bigger sister. And the small brother goes to her sister and asks her, Didi, artificial intelligence kya hota hai? Then what she told, if mom gets angry on something, without telling anything, dad will clean the vessels, cook and get ready for tomorrow. That is artificial intelligence. So only with the action. the other things are happened. We'll get the sessions here first. Yeah. We'll be calling, yeah? We'll call them first and then. ఇంకోటి పోస్టర్ ఇది Okay. Very good evening, the delegates, and we are at the end of our program, and the most awaiting event of the program, the valedictory, like giving the best poster and oral for each of the allotted halls, and the virtual posters or orals will be given within a week. The things whatever we have done physically or we are giving now. And before that, I would like to invite Haima ma'am, Dr. Haima ma'am, to take away the session chair certificate. I would also request Dr. Vijaya ma'am to come on to the dais to take away the certificate. Now coming to the best oral presentations. So we have Sundip Chaurasa. Sundip. May I confirm the title, Design, Development and Optimization of Stable Lyophilized Protein Formulations? Next we have Karuna Tirumarella, Ischemia Reperfusion Induced Myocardial Injury in Rats. Karuna. SM Vanamati. Do we have SM Vanamati? Jupali Prashant. I think he is from ICT. Shreya Ajay. Preparation and characterization of different polymorph and formulation of immediate release tablets. And next we have Cynthia Lizzie Lobo. And next we have Ayesha Muhammad Abdul Moin. Rajendra Ankita Singh. It's okay. Anyone can represent. Coming to the best poster presentations. Praful Vijay Kumar Shinde. Any of his friends also can collect. Praful Vijay Kumar Shinde, astonishing findings of 5 uracil. Avinash Wak, RPHPLC and HPTLC analysis of cuminoids. Any friend of Avinash? You are Avinash? Okay. G. Srinath. Pooja G. Akar. Pooja G. Akar. Kartika M. Kartika. I'll read up the college names later because we don't have college name in the certificate. Akash Annie. Akash. Anup Prasad Bankar. Anup Prasad. You are okay. Anybody from Sri Venkateswara College of Pharmacy, Puducherry? Went. Anybody from Amenity University? Yeah, some voice said that you will end with your voice. It's the time. Thank you so much, all the delegates. Thank you so much. We'll meet in the next conference, okay? Please, all please go safe. We'll take a group photo, please. Come. Karuna. Aruna Rale. ఏ కాలేజ్ ఎవరు అరుణ అన్న వచ్చిందా ఏమో చూద్దాం
उधर से मैडम उधर से ये बोलेगा निकल जाएगा टेबल है निकल जाता गिर जाता गलत गलत बोला